So when we have the stinging, then we dissect the aphid, and then we check if the egg is there. So then we are sure that the parasitoid female has accepted the host, for the offspring.
Yeah, so stinging, the lady is actually the parasite.
But if you use parasitism, then you combine both preference performance in a single metric. Then you cannot test any relationship. Then no good paper.
Okay, thank you, Lian. Thank you, Nico. So let's come to the second speaker. Let's welcome Dr. and Professor Ye Jian. You know that, let me introduce Ye Jian a little bit. Dr. Ye is super famous in China, even is still young. He's currently the director of the Lab of Agricultural Microbiomics and Biotechnology at the Institute of the Microbiology, CAS. And he also the winner of the NSFC Distinguished Young Scholar Funds. Okay, and he got his bachelor in Zhejiang University and then get the PhD degree so in CAS. And then he moved to the Singapore and to do the postdoctoral research in the Temasek and of the National University of Singapore. Everybody know in China, and he published a great paper in Science as cover in this April. And his research team published this paper and enriched the successfully elucidated, just discovered the core molecular mechanism of the citrus, Huanglongbing. I think this is, we call it cancer of the citrus. And actually, and this, the disaster plant disease cost billions US dollars per year in worldwide. And he resolved the long-standing challenge of this lack of resistance genes in citrus plants. Okay, you have 13 minutes.
Okay, thank you very much, Professor. So very warm and very nice introduction. So it's my great pleasure to share our data and our research progress in citrus Huanglongbing, and also in Europe country they may know it as citrus screening. So that I just talk with our Spanish friends. The name of my name is just Ye is means leaf, and Jian is means healthy, that healthy leaf. So that means my mother and my father gave me my name as my whole life, and to do some healthy leaf stories. So today I will go to talk about a little bit of our Huanglongbing, how we identify the target proteins which we can use for identify the therapeutic drug development. Since you may all know that my group heavily work with insect-borne diseases, including both bacteria and also the viruses diseases. In my lab, I mainly work with citrus screening and also the tomato, which transmitted tomato Gemini virus, which transmitted by whitefly. So all of those kind of insect-borne diseases are very headache for human beings, since that the pesticide is also a very big headache, and also the pathogen and very difficult to control. In that case, the host plant actually face two big headaches. In that case, the host almost there's no disease resistant or insect resistant genes. Therefore, the only ways to control those kind of insect-borne diseases is just spread pesticide. But as you may know, and many pesticide usage will cause drug resistance or drug tolerance. Therefore, the control efficiency is very, very low. And in past 20 years, I'm working with this kind of pathogens. Okay, so you may notice that this our science paper is actually very nice photos to show why the insect-borne diseases is very difficult to control. You may notice that this is citrus young shoots, and there will be insect eggs inside of inside of the citrus shoot, and also you will notice that the larvae and also the adult in that way. And so that for one generation, the salad, citrus salad can produce around one produce 7, even 1,000 next generations. So that once even a single pest did not killed by the pesticide, then the next generation, there will be another 1,000 comes out. Therefore, the disease control for this insect-borne disease is very difficult. So not only the citrus greening and many of for example, the Pierce disease in grapevine, and also the olive quick decline syndromes. Last year I visited Italy. There will be a very big problem for Europe people there. And all those kind of insect-borne diseases are no drug and no cure. Why? So that's the main reason for those diseases is that we cannot culture those pathogens, those bacteria. So for human, the diseases, people are trying very hard to find ways to culture them. And but for plant disease and plant pathogens, those kind of non-culturable pathogens, and will not so heavily research. Therefore, we require more effective effects, more trying to do those kind of diseases. For the citrus, I think that people will always think that it's a great gift from God to give human beings, and almost major countries in the world can produce these fruits, and it can bring you many nutrients, and also some perfume. Also use that the citrus extract as a perfume. Therefore, the value of this industry is very high, more than $100 billion per year. So that right now the citrus, the orange juice price is even more higher than five-fold price higher than 10 years ago or 20 years ago because of the citrus greening. Okay, the citrus greening or hornbeam is a major disease, and Professor Wang called it the cancer of the citrus. Yes, it's correct. So luckily that in Europe countries there's no citrus greening yet, but all of the other continents have these big problems, especially in China and in South America. These have exist. But so far, there's just 50 years ago, people get to recognize it is not abiotic stress, but it is a pathogen-caused disease. Therefore, this is the only disease named as the Chinese name, hornbeam, and the Bonville from the France, the same academia from the France as Nicholas. He is a very brilliant guy, and he realized that Actually, there's a Chinese scientist, Lin Kongxiang, actually identified that, recognized that it's a pathogen-caused disease. And there will be 3 species from this for the causing pathogens. And we developed a very nice PCR-based detection method for this disease. And the major causing pathogen is CLAS. That means the Asia species from this genus is a major pathogen in the world, all over the world. And 20 years before that, this pathogen has invaded into the United States and caused a very big problems for the United States. And it caused around, especially in the this Florida state, and around $9 billion every year loss. Okay, so the disease circle is like this, and this Asia citrus silage transmitted from the diseased trees and get infected into the new trees. And this pathogen is very special. Although it is a bacterial pathogen, but it actually, the very, very small size is only 1/10 of the normal E. coli size. It's around 200 to 300 nanometers. As you may know, E. coli in our gut is around 5 mils. Then it's around less than 1/10 of this. And the genome size is also very small. It's only 1.2 microbase, and only 1,000 genes of the genome. And there's no cell walls. Therefore, the traditional ampicillin antibiotics cannot kill that pathogens. Okay, in summarize, I will tell you that our work actually consists of 2 parts. First part, we identify the targets, which we can use for identify the antimicrobial peptide, which also can inhibit or kill the pathogens. And the second part will include that we use AI as very rapid ways to identify those kind of peptide, which we can use at a very good control efficiency under the field conditions. This is our AI programs to identify high efficiency antimicrobial peptide, and this is our target proteins, POP21. POP21 is our target proteins. So when 10 years before, then I'll come back to China from Singapore. So I sought to find some answers to solve the problems for all the citrus farmers. And China is the origin countries, the regions for the citrus species, also other relative or far relative species for the citrus. And some of the Daoxian Yeju is a kind of Daoxian is the Hunan province in south of China, and a very nice tolerant. It's a good tolerant to citrus springing, and also the osman. That means that it's origin from Australia, and it is quite good resistant to citrus springing. So we are doing the transcriptome analysis, and not only those citrus species, but we also did far related. That means the Jiulixang is jasmine orange. It's very resistant to citrus greening, and the curry plants is totally immune to citrus greening. So when we get those resources from all over China, so actually we identify, we collect quite a large number of the wild species from China and do the inoculation and identify which one is resistant to citrus greening. So with those kind of very big germplasm resistant to citrus greening, and we do the transcriptome analysis. Okay, these are our transcriptome analysis data, and actually, when I was a postdoc in Nanhai Charles lab in Rockefeller University, then his lab worked quite heavily on the protein degradation or the protein, how the protein control the disease resistance. So in our first analysis, we identified quite large number of the PAP genes. PAP means it's a kind of protein E3 ligase. Those E3 ligases have been, some of them have been identified as sensitive genes, sensitive proteins for the disease. In that case, we are very lucky to identify, we noticed that actually that quite large number of the PAP genes is very look like sensitive, disease-sensitive genes, not the disease resistance, as the opposite side of this. So among them, the PAP21 is the highest expression in sensitive plants. That means in the orange, normal orange plants, there will be very high expression. But in the immune-resistant plants species, there will be very low expression. Then we saw that there is a negative correlation between the expression, the gene expression, and the resistance profiles. So we use that as a candidate for a disease-sensitive genes. So we noticed that actually the expression of the PAP21 is quite negatively related to the HLB, citrus greening immunity. And when we do the cloning, we noticed that our graduate students, one morning they told me there will be a mutant. Seems that it always present in some immune, we cloned from the immune plants. For example, the curry leaf and the jasmine orange plants. Then I thought that probably it's some point mutation is just random. Why they always come to a random sites, which we identify that you can notice that okay, that the graph that there's a TCG coding as cysteine and GCC as calling alanine. So that it's only two nucleotide mutation, and they always present in wild species which is immune to citrus greening. So we have a hypothesis. Then the possible is some kind of mutation or the duplicate genes in those kind of immune plants. Then we cloned all of them and do the assays to the, so that cannot see that, do the enzyme, E3 enzyme assay. It's really, it is dominant negative genes, which that means when this mutant present in the citrus genome, then the citrus plants will be immune to the citrus infection. Okay, so we did the transgenic plants to confirm that data, that hypothesis is correct. So that when we do the transgene, we overexpression or expression those dominant genes in citrus, in common citrus oranges, then it converts the plants sensitive to resistant to citrus greening. Also, when we do the INI in the orange, common orange plants, then the disease resistance will come to very nice data in the left side of the plants. Okay, then we did the genome editing. Since that if we convert the resistant gene into the resistant genes, sensitive gene into the resistant gene, that's very simple. We just do the prime editing, genome editing, to convert a single amino acid from the cysteine to the adenine. Then we get a very nice resistant results. Okay, then next step, we identify these target proteins. So since POP21 is disease-sensitive genes, so we will probably want to try to identify some protein which can be degraded by this E3 ligase. So the E3 ligase, one of the target is MYC2. MYC2 is just morning acid pathway which have been well known to resistant to the insect herbivores and also disease. So that we think that is a very good target for degrading. That means sensitive genes to degrade the resistant genes. Once the resistant genes go up, that means the sensitive genes go down. That means if resistant genes lower, that means the sensitive genes go up. We do the biochemistry data. Well, we'll not go to detail. And look at these pictures. We know that the CS, this means our orange, widely used the citrus, and those curry leaf and the jasmine oranges, the MYC2, that the resistant genes go very high, and these orange common cultivated orange plants, that the MYC2 expression or accumulation is very low. Okay, then we identify the whole pathways, which means POP21 as sensitive genes, and the MYC2 as resistant genes. And with this circulate, it controls the just morning acid mediated disease resistant and the pesticide, the citrus salad resistant. Okay, so for the genome editing, that we need quite a long time to come to the market for people to use to control the citrus resistance. So we identify some of the small peptide, both from the human gut and also the reservoir. One of peptide we write very heavily is come from our human gut. Almost all people have this peptide in your gut. So that means there will be not so much caution or so much worry about to use those peptides for the citrus, because you always to eat the fruit, right? So that we need to be very careful to choose which peptide we should use. And one of them we call the APP3. APP, that means anti-protein proteolysis. That means we call the APP3.14. That it means apple pie. We get one name for this peptide. We call it apple pie. This apple pie is really work very nice to control the citrus graining pathogens. And we use that for this small peptide that is solved in the water and inject to the junk injection into the citrus plants. And after 2 months, we inject our peptide that the new shoots comes out from the treated plant, citrus plants. This is our greenhouse. And after 6 months, you can look at the citrus titer, citrus titer trait After 6 months treatment, that means the CLAS, that means citrus greening pathogens have been totally cleaned up inside of the plants. Not only the leaf, but also the roots. Because that the root part also can get a very high amount of citrus greening pathogens. And we can clean up all the all citrus greening pathogens inside of after 6 months treatment. After we remove that treatment, then the pathogen cannot be recovered. That means we have already cleaned up all the pathogens inside of the citrus. Okay, not only the pathogen, but also the apple pie also can work with the insect vector. Actually, very interesting. So most of the insect-borne diseases, the pathogen can control the pest, the vector preference. That means to help the virus or pathogens to transmit into the new plants. That their strategies. Okay, so that's a new papers just come out in the next last week. We found that actually our apple pie can clean up the pathogen inside the vector inside the insect. Once they take the peptide in their body, and the pathogen can be cleaned up not only in the plant side, but also inside of the salad. And also this apple pie can convert citrus repellent to the salad. That means not only can reduce the pathogen itself, but also can inhibit or reduce the vector insect to prevent to the citrus trees. Okay, we have done 3 years field trial in China, especially in Guangxi, Zhejiang, and Jiangxi. And our students did a very good job. Actually, it's a very tough work. And that you mean that you may notice that our treatment can make a very good results in the field under the field. Okay, so this year we see a very good international collaborations together from the citrus from Brazil, and Antonius is the director of the from citrus, and they fly to China, and we get the symposium to discuss the possible collaboration to use that small peptide in Brazil. Next month, I will fly to Brazil to test our peptide in Brazil and also in United States, Florida. Okay, so with that, I think that we also, because the peptide, right now we use the chemical synthesis, but we want to get the more cheaper peptide. Therefore, we use the manufacturing by yeast, and also by E. coli, by the bacteria, which we can get the biopeptide from very cheaper ways. Right now, we can generate those small peptide from our yeast and E. coli fermentation processing, and we use that in citrus. It's already for 2 months and 3 months. And it worked very nice. Okay, with that, I will going to end here, and I will thank all my labs, and thanks to all our collaborators all over China, and we also get the kind of help from Daniel from University of California, David. Okay, thank you very much.
Okay. Thank you, Professor Yuan. It looks like it's amazing small peptide. And I also think that already win the respect from the scientific field. Maybe in future he could become the billionaire because he controls the the medicine to kill this cancer. Okay. So, are there some questions or comments you want to negotiate with? Okay, thank you.
Thank you very much for this incredible work. I have a very simple question. How do you apply the peptide in the field? I saw like a kind of bottle. Do you inject the peptide or do you treat it?
Yes. Okay. So it's a very good question. Actually, in China, people already use some kind of this injection method. They use antibiotic actually in the field, but it's not allowed to use antibiotic. Especially the antibiotic is human use or animal use. Then in that case, that would be very problem for the drug resistance or so that we use very small bottles and to the injection into each big trees. And right now we are also trying to solve that problem with our fermentation formulation adjustment. We can use that spread into the leaf spread and also the water, the soil spread. Yeah, a soil spread on the leaf spread can work a little bit, but the intake efficacy is very low because that we need to improve that. Yes.
And a second question, please. Have you already treated with gene editing in citrus? Do you have any result on that?
Genome editing. Yes, we actually there will be another paper we're trying to send out for review. So we have this genome editing trees for resistance, for the disease resistance. Yes. Yeah.
And can you advance some results on that?
Yeah, it's good, quite good. Yeah.
Okay, thank you very much.
Okay, thank you. Are there more? Oh, yeah.
Thanks for the talk. Not clear to me what you did with the vectors when you said that you clear with the peptide the pathogen inside.
Oh, okay. So due to the time limitation, I didn't explain that part. Okay, you mean that? Yes. So once we inject the small peptide, then we can increase the citrus resistance or because that actually in my lab work quite heavily with this kind of volatile controlled repellent to the pest or to the insect vector. So once the citrus screening infected, the volatile of the citrus will be totally different. So therefore, the repellent will be reduced. Then we use the peptide to control that, to reduce the reduction of the repellent. Then the citrus plants will quite resistant to the insect in the water from the volatile side. Then another data we didn't publish is once the salad was infected by the pathogens and the carrier pathogens, the pathogen can multiply in the insect vector. That is a major reason why the insect-borne disease is very difficult to control. In that case, once they intake one pathogen, and it can produce more than millions or billions of pathogens inside the. Then with our peptide injected the citrus junk, and the insect can take by inject, right? So then the pathogen can be killed also inside of the insect. Okay, that makes sense. Because our peptide is actually some kind of antimicrobial peptide. Yes, it's a multifunctional peptide.
Yeah. So there's a last one. Yeah. Okay. Or maybe
Thank you very much for your talk. My question is about how long this protection of both the plant and the vector. So, is there any chance to reinfect the same plant or the same vector?
Yes, understood. Okay, it's also very, very, very nice question. It's actually we try right now. We use triple times every year for citrus. We didn't exactly know which is the half-life of this peptide. Right now, my graduate students still take very, very detailed analysis on this peptide. But you may know that when people use the snake root as a weight control for human, the native GLP antigens is only minutes of the half-life. But right now, the product our human use that peptide, right? So it around 2 months, the half-life. So that quite large of a space for improvement of this peptide performance. Yes. Okay.
Okay, Felix, you want more?
One final question then. With regard to the application in the field, do you, what you presented here is curative, I understand. So you have citrus trees that are diseased and then you treat them. Do you envision this as being also used preventatively?
Yeah, can be increased plant resistant or repellent to the insect also. And I think the last question just I mentioned that the peptide still have a half-life. So if you inject earlier, and it still can work, but you need to inject more times. Okay.
Okay, thank you. Also, time is up. So if you have some questions, you can communicate with Ye during the dinner time. Okay. So we have 3 remain, huh? Let's welcome the next one is I just checking the dictionary. The pronunciation is too hard. The Mikhail Recupero, is that right? Okay, Dr. Mikhail is an assistant professor in applied entomology division at the University of Catania, Sicily. Came from Italy. And he works on the sustainable controls and the insect pest of protected crops through biocontrol, biopesticide, and using some techniques. So today, I think that he will mention the story from his lab and around the severe, pest on the cotton. You have 30 minutes, okay?
Yes. Thank you, Professor Wang Su, for the excellent organization of this conference, and thank you all for attending here today. Well, it's my pleasure to present the IPM tools that we develop around to control Phenacoccus solenopsis. Doesn't work? Well, as you know, climate change exacerbates the worldwide problem of invasive species that are fostered by the intense exchange by humans. And these cause several ecological challenges, especially in agro-ecosystem, because ecological communities are disrupted. Among all these challenges, Phenacoccus solenopsis, also known as the cotton mealybug, it's species that is becoming invasive in the Mediterranean basin recently. Actually, this mealybug can be recognized by the presence of black bands on the dorsum. However, this species can be easily misidentified by other similar species that also attacks solanaceous like Phenacoccus solani. However, the identification can be done by experts through specific morphological keys that focus on typical morphological features on the abdomen of these species. However, hopefully, molecular diagnostic and in particular these two primate species can help us to identify this pest. If we look at the geographical distribution of this species, it is native from North America. Indeed, this pest was first described in the late 19th century by Tinsley in New Mexico, and then the pest spread through Americas the last century, and then at the beginning of 2000, it was reported in Asia, where it became a pest of cotton, especially in India and Pakistan. Then, in 2020, the species was reported in Europe, in particular in 3 countries, Cyprus, Greece, and Italy, where we reported for the first time on tomato, and then spread among other Mediterranean countries, including Tunisia, Algeria, and so on. We conducted phylogeographic analysis, and apparently the population we retrieved in Italy and also in other Mediterranean countries like Tunisia belong to the same clade of Asian Achesian. It means that a possible introduction of this species is due to infested commodity. If we look at the host range of this species, more than 300 different host plants belonging to 65 different families are hosts of this pest. In particular, also glasshouse vegetables, solanaceous plants like aubergine, tomato, and sweet pepper, and also ornamentals. This pest is also a problem for ornamental plants like lantana or hibiscus plants. The species can attack different plant parts like leaves, stems, but also root collars, and with this typical infestation. It reproduces by sexual reproduction, but also facultative parthenogenesis can occur. And this pest has a high reproductive potential, and the cycle can be done in less than 1 month, and consider that there can be overlapping generation up to 12 every year. And it's important to note, as for other mealybug species, of course, there is a mutualism with ants because of the honeydew production. And for this species has been highlighted a mutualism with the red imported fire ants, Solenopsis invicta, that is an emerging pest. And we recently recorded in Italy 2 years ago, and there is, of course, an eradication program. And it's important also to study the interaction with ants, especially for their spreading. If we look closer to the damage of Solenopsis mealybug, as you can see, it causes weakening, distortion, chlorosis that are associated to the direct subfeeding and the producing of the honeydew. And in case of large infestation, especially when tomato transplanting are done, this can cause dieback. This pest is generally controlled by insecticides in India and Pakistan. Indeed, some chemicals are authorized. However, in the EU currently, even though the EFSA report as a potential quarantine pest, chemicals are not allowed in Europe. Indeed, farmers control it often with good in controlling the pest. If we look at the essential oil, only garlic and peppermint were the most promising botanicals in controlling this species. And essential oils can be promising, even though the lethal concentration we estimated was quite high. It means that we have to improve the formulation. Then, as you know, it's really important again, chemical insecticides like sulfoxaflor or thiamethoxam significantly reduce the survival, while acetamiprid and pyriproxyfen also alter the behavior of the bumblebees. And therefore, some improvement need to do, and especially the selection of chemicals in integrated programs should be done. Which are the future direction of our work? Well, we are conducting other efficacy tests of selective synthetical and also botanical chemicals on these pests by testing other extracts. For instance, we have a collaboration with a Spanish university that provides us guanyule extracts. That is a plant where you can extract also rubber, and it's also good for the production of different active ingredients like argentatins that has insecticidal activity. Also, we are focusing efforts on studying the indirect effects that microbial can as plant defense inducer, because microbials like Trichoderma or Bacillus, when administered to the soil, can improve plant defense and then trigger indirect defense on T trophic network, and as an effect on both pests and their natural enemies. And by the end, we are evaluation the biocontrol efficiency of biological control agents in the field that should be assessed. And also it's important to improve conservation biological control in the field by the use of anchor plants. We're testing for other studies, for instance, verbena or sesame plants that are already used for improving other natural enemies, in particular for the control of tuta absoluta, but we can also translate to these systems. I want to thank the big group of the University of Catania I belong. You can have some information by this link, and I'm available for your question, and thank you.
Okay, thank you. Thank you, Michael. You just show us a very classic procedure to evaluate a new invasive species and create the package of IPM tools, and to show us how it has worked. So like Nico, we run the Adopt IPM project D. So there are some questions. It's very clear, the simple work. So I think you're ready to tell them everything we want to know. Are there some questions? Okay, if no, okay, there is a guy. Oh yeah, Yangfeng.
So thank you for your detailed presentation. So I have one question. I noticed that you use essential oils to control the cotton milling bug.
Yeah.
And do they cause any harm to the host plant?
Very good question. Yes, we at the beginning of our experience with essential oils, they cause phytotoxic effects on plants if you use as crude extracts. But in this case, we develop nano formulation by the use of the essential oils and the percentage of 15% almost. The use of a surfactant that basically is a soap and water. All these three elements were sonificated by the use of sonification process that creates an emulsion. Okay, so we spray plants with this emulsion that has no phytotoxic effect.
Great.
We also develop. You can see in our published paper, there is also a phytotoxicity index that help you to evaluate any phytotoxic effects on plants.
Yeah. Thank you.
Welcome.
Is this essential oil expensive or no?
Well, it depends. Some of them are quite cheap. For instance, citrus essential oils are quite cheap because of the citrus industry. Same for garlic, while for other, for instance, Artemisia essential oils can be quite expensive. Also because some essential oils, as you know, are used for pharmaceutical or for aesthetic purpose.
Yeah. Okay. I think I'm afraid maybe some as expensive as a perfume. Essential oil. So thank you very much, Mito.
Thank you all.
So let's welcome Felix Walkers. Professor Felix has worked in the field of insect plant interaction for more than 30 years. And now he is a position like the leader of the R&D director of the BioBest. Actually, he published so many papers. I checked last night. Even I drink a lot of wine, but I checked everybody's publication. So I think that Felix did a lot of amazing work. So you have 30 minutes to introduce what you did and welcome to
okay. Thank you, Professor Wang, for the kind invitation for me to come over here and participate in this very interesting symposium. As you alluded, I started off in academia, then I moved to BioBest. And from my experience working at a biocontrol industry doesn't have to be so different from academic work. So we try to continue to publish and to work in the Today I will give you an overview on the trends and innovations in biocontrol with a focus on arthropod-based strategies for biocontrol solutions. First, a bit of introduction on BioBest. You have heard the name of our company a number of times, also thanks to the collaboration that has been signed today. BioBest is a worldwide operating company in the field of crop pollination and crop biocontrol. We are active in all continents, and we are present in more than 40 countries, including China, where we have been working for several years now. We have also 250 distribution partners, more than 3,000 employees, and their headquarters is in Belgium. Our products are bumblebees for pollination, and that's where the company started. Then with the production of bumblebees came also the demand from the growers for biocontrol solutions, because if you use pesticides together with bumblebees, it's not very compatible. And so very soon after the introduction of the bumblebees, we diverged into the area of biocontrol. And we started in 1987. Our products are beneficial insects and mites for biocontrol, also biopesticides. Technical advice is a key part of our business. And we also have been branching into high-tech and artificial intelligence, for instance, for automated monitoring of insects, insect pests and insects, natural enemies. The R&D department, which is the department that I'm responsible for, 32 researchers in 5 teams, focusing on these different topics that I mentioned before. And with BioBest R&D, we have a number of roles. We developed new local species for pollination and for crop protection. We also look at new strategies for biocontrol, and then we have a number of tasks, including interactions with academic groups and interactions also with regulators when it comes to registration of products. If you look at the microbial biocontrol, and we already heard from the talk from Nico that in classical biocontrol, it's vital that we have specialized biocontrol agents, and that's also in augmentative biocontrol, so the mass production of biocontrol agents and the release. The specialists also play an important role. But in addition, we have seen over the last few years or over the last decades that generalist predators are becoming more and more important for augmentative biocontrol. And the advantage of these generalist predators is that they act against several pests and pest stages. Some of them even control pathogens, as I will show you. There is no need or less need for synchronization. They are easier to rear because we can use alternative prey and alternative diets. They can be established before the pest is present and feed on other food sources, including plant sap. So they are sometimes plant feeders without necessarily doing damage on the plant, but having the advantage that they can induce plant resistance. So you have a double-edged sword in that case. And as a result of the fact that we can establish them preventatively, we can use them as a standing army. So having them be present before the pest arrives. So some examples of a number of microbial insect predators that we have developed over the last several years. We have put some hoverflies on the market for aphid control. Then we have Mycomus, which is a brown lacewing, also for the control of aphids, where the adult is also feeding on the aphids in contrast to other green lacewing. We have Chronomatus ubiquitous, that's this mite that you see on the left, at the bottom left, which I will introduce to you later on. And we have the predatory trips. And the last two examples are examples of biocontrol agents which were not used previously. And it shows that there's still a lot out there in terms of developing new biocontrol agents. There's whole groups of biocontrol agents that we have not been looking at, and we can develop those for particular solutions in crop protection. So in addition to looking for new biocontrol species, we also, as I mentioned, are looking for new strategies, and I always use this example here to show you how these strategies can work. So what we try to do is develop tools to make our biocontrol agents that we have perform better. And you can think of Usain Bolt here, which we know is very fast. But we have this man on the right, which is actually faster than Usain Bolt, talking strictly on muscle power. And the reason why this man is faster is he has the right tools to use his muscle power more efficiently. And that's exactly what we try to do with these new strategies as well. We use our biocontrol agents and make them more efficient. One of the ways in which we can make biocontrol more efficient is by providing resources that these biocontrol agents need. And if we look at a modern agricultural field, that's here an example from Europe, but the same would apply in China. The field is very clean, the fields are getting bigger and bigger, and it means there is no resources other than the cropland present. And a lot of insects need floral resources to survive and to perform. We know this for pollinators, that they are associated with flowers. Not everybody knows that the same applies actually for biocontrol agents. Also, biocontrol agents are, in general, dependent on floral resources to survive. So we can resolve this situation by actually reintroducing floral resources in a controlled manner by using these kind of flower margins. And we'll hear more about these flower margins in the talk this afternoon. Just to give you an example to show you that this is not a trivial issue. This is a study that was done by my PhD student Karen Winkler in Wageningen. And what she did was she worked on cabbage as a model system. She had large cages in which she introduced cabbage plants. And on the cabbage plant, she introduced caterpillars of the cabbage moth, which is also a pest issue here in China and worldwide. Then she introduced a single parasitic wasp that attacks this cabbage moth. In half of the cages, there was no floral resource present. In the other half, a single flower of buckwheat was added to the cage. And then she looked at the performance of these parasitic wasps. What you can see at the bottom, the two bottom lines are is the parasitism by the parasitic wasp without the buckwheat flower. You see the parasitoids survived just a mere 6 days and attacked maybe 4 or 5 caterpillars during their short lifespan. In sharp contrast, by providing this one buckwheat flower, you can see that the parasitoids lived more than 30 days and they attacked more than 300 caterpillars during this lifespan. So it has a tremendous impact if these floral resources are being provided. So you may say, well, this might be a a selected example, but here is a list of all biocontrol agents that are dependent on nectar or pollen, so floral resources, to survive and reproduce. And everything, almost everything that we use in biocontrol is on that list. So the use of floral resources is really a key element to make biocontrol work. So if we want to go and select these flowering plants for our flower margins, how do we go about this? How do we optimize these ecosystem services? So we start with the presumption that different organisms have different needs. So not every biocontrol agent has the same requirements, and pollinators and biocontrol agents have quite different requirements. So going for flowers that are suitable for pollinators doesn't help your biocontrol. So one take-home message, it's not a one-size-fits-all solution that we're looking for, but instead we're actually going for the tailor-made approach. We tailor our flower margins to the requirements of biocontrol agents. And these are the kind of studies that we have used to come to the selection of flowering plants. You see here a range of flowering plants organized according to the depth of the flower. So the top ones, the nectar is very accessible. The bottom ones, the nectar is less acceptable. And we see that the biocontrol agents are really restricted to the flowers that are very open in their nectar accessibility. So what we do is we select the flowering plants that are suitable for all major groups of biocontrol agents, and on that basis we create these flower margins. This is something which we then have taken from the lab to the field, to the farmers. One example is this project in the Netherlands, south of Rotterdam, where we have started in 2004, so more than 20 years ago, a project in a larger area where we introduced these flower margins. By now, 20 years later, 50% of the farmers participate in this project. They have taken the project on themselves. They have started a cooperative to continue this because they are excited about the results. We have now more than 500 kilometers of flower margins in this area, so it's a beautiful area to drive through as well. And what most important, by using this approach, we have shown that we have achieved 90% reduction in pesticide use by these farmers compared to growers in the same region that are not participating. So it shows you that this method really can work. We have then rolled this out to other countries. So we have a number of projects in Germany, UK, and in Belgium. And this is something which really is taking off, and I think as part of an IPM toolbox, very important. Food supplementation, so you can do this by using these flower margins, but you can also take another approach. You can also say, can't we provide foods directly in the crop? Can't we spray foods on the crop in those cases where flower margins may not be possible, or where the biocontrol agents are not mobile enough to move back and forth between the flower margin and the crop? And there you can think of a number of different possibilities. You can use pollen, you can use sugar water, you can use alternative prey sources like Ephestia, Artemia, or prey mites, and you can even think of the rice flower moths that we discussed yesterday that could also be used as a supplementary food in the crops. And artificial diets is another possibility where you create a diet and apply this. We have started about 12 years ago with the approach of providing pollen as a commercial product. So growers can buy pollen, blow it on their crop, and by doing so, they can support predatory mites and other biocontrol agents like predatory ticks. We use pollen from Tifa, by the way, which is a plant which is growing worldwide. It's FIFA pollen is also available in China because it's used in Chinese medicine. And here we see how it works. You see the pollen on the crop. You see the predatory mites feeding on the pollen and multiplying just on the pollen. So there's no pest available present here. The same for hoverflies and the same for predatory pests. To give you an example how this works in the field situation, here are some studies we've been doing in onion, where there's a big problem with onion trips in Europe. A lot of pesticides are being used to control this onion trips. And what we have been doing is go to a conventional onion grower, ask the onion grower if he can have part of his crop where he would not spray, and the rest of the crop would be treated conventionally, meaning spraying 3 to 4 times. There's also a flower margin next to the onion crop, and then we apply the pollen on the part that is not being sprayed. Then I did monitoring of the onion trips population, but also of the natural enemies in this system. Natural enemies being mainly lacewings, spiders, and predominantly predatory trips. And what you see is that if you provide the pollen, you get many more biocontrol agents, both for the predatory trips and for the spiders here. That's partly because they can benefit from the food, the pollen, and partly because there's no pesticides being used here. And if we look at the impact on the onion trips, to the left, we see the plots for the pollen treatment, and we see that over the year, each bar represents a monitoring time point over the year. So we see over the year that in the pollen treatment, the number of onion trips, this is per 10 onion plants, so it peaks at about 5 onion trips per onion plant. And then it actually declines. Even without the use of any pesticides, the onion trips population goes down. In the control, where the pesticides are being used, we see that initially the onion trips is very well under control. But the 2 last monitoring time points show that with the absence of the predators, the onion trips is actually reaching much higher populations. And just before harvest, there were 67 onion trips per onion plant. And this is then how it looked at the time of harvest. You see the brown parts of the field are the parts which had been treated chemically. The onion trips got out of control, and there's a total damage on the onions. In the right corner, you see the part that was not treated chemically, but was treated with pollen. The natural enemies have done their work, and the onion crop remained green and viable. And we did do some harvest assessments, and we see that the yield in the pollen-treated part was 8% higher, and the weight of the onion was 15% higher as well. So it's a dramatic impact by replacing chemical pesticides with this approach of feeding your natural enemies. This is something we repeated over 4 years. Each of these 4 years, we see the same results, same good results. And this is how it looks like in the field situation. So this is the data from this year, which is interesting because this year was an extreme high trips pressure year. And we see that even under those conditions, the pollen plus in this time the release of the predatory trips yielded again much better results than the chemical treatment. So what we see here is what I call integrated biocontrol. So we use various measures that we can use of biocontrol agents and conservation biocontrol to support those biocontrol agents. And what we have here is we have the flower strips, which is a good source of predators, including the predatory trips. The second pillar is that we use the pollen to feed the biocontrol agents in the crop. So you also lure your biocontrol agents from the flower strip into your crop. The third one is that we do scouting. Monitoring and scouting is an essential element in successful IPM, but also in integrated biocontrol. And the fourth is the release of predators, or in those cases where there's not enough predators available naturally. And this is with these four pillars, you have a very robust system for your biocontrol. I already mentioned that we had this mite that was effective against a pathogen, against powdery mildew. I would like to share you a bit more about this new group of predatory mites. So it's not a picoseid, it's a tidaeid mite, it's a different group. And we developed this mite to control tomato russet mite, to control a pest, which is what you would do normally with a predatory mite. And when we were doing these trials, we saw that the plants were very well protected against the tomato russet mite, but we also see in the right that the plants were completely clean of powdery mildew, whereas the control row, which was right next to it, was full of powdery mildew. So there was a high pressure. And this showed that this predatory mite is actually able to control powdery mildew, on the one hand by feeding on it, and on the other hand, as we have demonstrated, by inducing the plant resistance, because this mite is also a plant feeder. It induces the plant resistance, and as such, the plant becomes resistant to powdery mildew. We see the same in other crops. We've seen it in cucumber, strawberry, and even in vineyards. So here, in strawberry, a lot of chemicals are being used. Strawberry is seen as one of the most chemical-dependent crops. And we see a conventional grower is using about 12 times fungicides against powdery mildew. We compared that with our mite, which was released only once, fed with the pollen again. And we see that without any fungicide use, we have much lower mildew pressure. And on the fruits, it's even more pronounced, this difference. So we can replace 12 pesticide treatments with one release of a biocontrol. Then going back to the previous speaker, who already mentioned the ants and the interaction with mealybugs. This is another topic that we have been working on, where honeydew is used as a sugar source, and honeydew is actually often a problem fruit. The ants go to the honeydew and collect the honeydew, and then the ants protect the honeydew producers and chase away the natural enemies. So even though ants are very good biocontrol agents in and of themselves, they are often seen as a problem because of this interaction with honeydew producers. So what we wanted to test is the alternative to the use of pesticides by distracting the ants from collecting the honeydew. So if we produce or offer the ants an alternative sugar source, then the ants can be distracted by this alternative sugar source, and so we put the sugar away from the honeydew producers, and the ants will actually move towards the sugar source, ignore the honeydew producers, and the honeydew producers are then open to attack by their natural enemies. It's a very simple concept, but I think just due to its simplicity, it's also an interesting example. Here, the kind of bags that we have been using, it's a sugar feeder with a wick. There's a little grid on top to keep bees and wasps away. And then if you put that in your tree, you see all the black dots on there. Those are all ants feeding on the sugar. They really like it. We've done trials in a number of crops in citrus here, where we looked at mealybug control. And what we see on the top graph is that if you have the sugar feeders, that's the green line, the ant visitation of the mealybugs drops almost instantly. So they ignore the mealybugs, go for the sugar, whereas in the control, the ants stay on the mealybug colonies. And if you look at the parasitism rate at the bottom, you see that in the situation with the sugar feeders, the parasitism rate is more than doubled. And this was actually work that Alejandro Tena also did together with us. Here, the parasitoid that is doing the job in this particular case is Anagyrus pseudococci. We've done the same work in vineyards over several years. And what you've seen here is that in these different vineyards, those are 4 vineyards, we see the same consistent results. Every time when we put the sugar feeders in, the mealybugs are very effectively controlled. Keep in mind that this is a log scale, so we see a reduction here of mealybugs by a factor 1,000, just by adding the sugar feeders. Works also against aphids, when those aphids are untended. Here's some work from the Netherlands, where we have been looking at the rosy apple aphid. Again, the standard situation without the sugar feeders, a lot of problems with the aphids. On the right, situation with the sugar. And the biggest part of the graph is the green bar, and the green are those aphid colonies that have been eradicated. So we see that almost all aphid colonies have been eradicated using this method. And with this example, I would like to finish my talk, and thank you for your attention.
Okay. So some products will bring a loss of, that products, solutions, and maybe will benefit our Chinese farmers in future, such like this non-crop flowers. I did with Nico and with my group already for a decade around this. So more and more Chinese farmers already accepted, to plant this kind of flower and nutrition plants around the crop field. It's really amazing. And are there some questions around the Felix speech? It seems like you represent very clear. So I hope that my institute will have a good future, and in collaboration with BioBest. Okay, thank you very much.
Okay, thank you.
Okay, finally, the last one. That let's welcome the Professor Zhang Liansheng that came from the Guizhou University. He also is a close friend of me, and actually Liansheng Zhang is a co-leader with Nicolas Diniz around the Adopt IBM program in the China side. And Liansheng Zhang is super famous, and in China he spent almost 25 years and just focused on the periscope, especially the trichogramma. And so let's welcome. You have 30 minutes.
Okay. Hello, everyone. Thanks, Professor Su Wang, give me the chance to introduce our teamwork. So in this morning, so we can see most the talking focus on biological control with natural enemies. So yeah, so for this, for my talking, also focus on biological control with natural enemies, but this one targets different crop. So today I will give a presentation on development and the demonstration of trichogramma with biological control for the soybean pod borer. So now I just give a simple background. So why we conduct this work on biological control of soybean pod borer with natural enemy. So from this map on the soybean plantation, so in China. So China is the world fourth largest soybean producer. So with cultivate area of 10 million hectares in last year. So the northeast province from half China's soybean production. Among them, Heilongjiang province plants 5 million hectares annual, so per year. So from this map you can find, so in northeast China, so have the more than half the soybean plantation. So here, so we also have the plant come from Brazil. So Brazil have, I think, higher the soybean plantation than China. So for the soybean plantation, have lots of the pest damage. So soybean pests, so also have some aphids. Also have some caterpillar. So here's also two caterpillars who damage the soybean. So today I just focus on this pest. This is a small moth, the soybean pod borer. So this one is the most economically important pest in soybean production, especially in North China. So this pest just damages the soybean pod with the larvae. Also, we can give some introduction. So for this soybean, it's an important crop. So in China, so this soybean have economic the significance. So provide the protein, and we eat it as oil, especially in northeast China. I don't know what happened. So yeah, so for this the target pest, the soybean pod borer. So the traditional control method is use the chemical insecticide. So also spring, just during this past occurrence. So we spring chemical. Also in the past, we also use the chemical to kill this pest with the fumigation. So now we also have other methods, such as they wipe the sex pheromone. But I think for this method, it's a good way to monitor this pest.
We will need to change another slide. It seems like there's some code problem.
Okay.
Wait a minute.
But you know, in China, northeast China have a lot the soybean plantation. So it's not easy to do the operation to distribute the sex pheromone. Yes, that's okay. Yeah, so in North China, so have other two main crops. So the maize and the rice. So also the borer for the is the corn borer, and the rice stem borer is the main pest that damage this crop. But in North China, we have successfully controlled this pest with reduced trichogramma. So here, so the figure we can find. So yeah, we published this paper annual review of entomology with Nico and Wang Su. So from this figure, we can find the area under control with reduced trichogramma reached 4 million hectares only in 2016. So this data that provide this MOA. So for that's now we mentioned to control the cord, the borer. So for the rest, stem borer. So also our team developed a new technology that's the mixed release both trichogramma species to control this pest. So we can find, so since 2014, so the accumulated application area reached 200,000 hectares. So how about for this soybean pod borer? Can we use the parasite, especially trichogramma, to control this pest? So from this picture, we can find, so for this, the pest have different character to lay eggs, different from the corn borer and the rice bran borer. This pest so distribute the eggs so individually on the soybean pod. So if we want do this work, so control the soybean pod borer, one thing is important, we should get the natural enemy resources. So for this pest, I think this pest have a different character. So this pest have a casual diapod, no weight culture in the laboratory. But if we want to do this work, we conduct this, collect the natural enemy resources. So we just during the occurrence of the pest, adults, so occurrence, so we collect the adult, such with the sweep knife. So what's the objective? Just want to get the pest egg on the soybean pod. So we introduce this the eggs in the soybean field. So every 7 days later, we collect them back to laboratory. So finally, I check these eggs whether parasiticized by the active parasitoid. So finally, we can find the eggs parasite were collected. So it's important we should know who is this the species. But because this Trichoderma have a minor size, it's difficult to do the identification. But we just under the assistance from Professor Su San Huang, so we know this the active parasitoid. So we do this work in Jilin province, also in Heilongjiang province. So in Jilin province, we get this Trichoderma species Chilonis. So in Heilongjiang province, we get two important species, Leucaena and Australia. For both these species from Heilongjiang province, so it's firstly caught in North China. Second thing is important because now we have 3 species. Which one is the best one? So we do the screening experiment. So first one we do, because the Trichoderma is an active parasitoid, so we test performance of this species on different the soybean pod borer with different age. Yeah, from this data we can find, especially for 3 and 4 days X, one species, Leucaena, comparison them, have a good, better performance than other species. So from this data, we know, the Trichoderma Leucaena, so it's best. We also found the development on different age, the X. So the Leucaena and Australia can finish development after parasitization. But Chilonis do not finish development on 4th days, the soybean pod borer. So we also do another evaluation. So we just test the functional response of this species to soybean pod borer eggs. Yeah, from this parameter, we can find the Leucaena also have the has instantaneous attack rate. So we also conduct another evaluation. So we test the life table parameters and the parasitic characters of Trichoderma on soybean or borer eggs. So I think that for this life table parameter, so also it exhibits the Leucaena have the largest intrinsic rate of natural increase. Yeah, just based on this result, we can give a conclusion. Which one is best? Leucaena, followed by Australia and Chilonis. So now we know which one is best. So another thing is important. We should to produce this natural enemy with low cost. So But for this work, we divide two products. So one, multiple wasp species per host die. Another one, we produce this the Leucaena with a new intermediate host. We know the Chinese oak silkworm is best intermediate host. This we call the large egg. Why? In China, this intermediate host is popular to produce Trichoderma, especially in northeast China. But this host only produce two Trichoderma species, one Dendrolyme, another Chelonis. So just now I mentioned why in northeast China we release so much, the Trichoderma to control large, the area of the corn and the rice stand borrow. So just we produce this species to control this pest, because this the intermediate host can produce the Dendrolyme with a very low cost. But just now I mentioned that we have found three Trichoderma species. Two is a new record species. So we also test this species, the performance on this large egg. So we can find Australia also can parasitize this large egg, also have a high hybridization, but no adults emergence successfully. Leucaena also can have a high hybridization on this intermediate host, but only a few wasps can emerge. So that's because based on this result, we do, the new, we develop the new products. Because this both species have a low emergence, we do, I think, one interesting experiment, so co-parasitism with, this species with Dendrolyme and the Chelonis. So we can find this result, so co-parasitism this intermediate host by Australia and the Dendrolyme. So just now we saw, if we only introduce the Australia, no adults emergence. But from this result, we can get 70, just based on the co-parasitism. So we also have other combination. So the Leucaena and the Dendrolyme. So we can produce 27 per X. So we also test a different intermediate host, rather the species, Chelonis, and the Leucaena. So we get the different result. But in all co-parasitism combination involve Trichoderma right from large host egg, the parasitism on this large egg, so exceeded 75%. The highest emergence rate was achieved by the Leucaena right on the large egg and Chelonis right on the large egg. So just now we found the Leucaena have a low emergence on the Chinese oak silkworm, but we're looking for other intermediate hosts. So we found this Irish silkworm is a good one. So because this the large egg host of Irish silkworm, no dipole, no also distribute in worldwide, not like Chinese oak silkworm. Oh, that's a natural resources in North China. So here, so the life cycle of this the species. So the total cycle life cycle near 60 days. So we use this egg to test the performance of Trichoderma species. So why we try this species? From this figure we can find, because this Irish silk have a thin cell, the egg cell, so then the Chinese oak silk. So also I think for this species have other advantage. So in the development, the advantage in the development of Irish silkworm industry in South China. So now I work in South China, so I found this species, the hosts have two advantage. One, climate adaptability. So each year we can culture this species 6 generations. So another one, so the plant, host plant, this masang, so natural resources abundant in West China. So we can find from this map of China, so have abundant distribution in China. So now we also culture this species in Beijing province. So now, since the last year, we have established the Irish silkworm breeding bases in 3 townships. So you just utilize this host plant. So now we can culture this species. So every year, so provide the large intermediate host. So we also want to try, so which status of this Irish silkworm suitable to produce the Leucaena. So we test 5 status the host. From this the data we can find, so Leucaena achieved the highest parasitism rate on manually extracted, unfertilized, and the worst eggs. So we also find, so on different status host egg, so have different emergence, but we can find, so a little bit, low the emergence, so less 50%. So finally, we get a conclusion. So we use the MUW status X to produce the Leucaena. So another experiment, so we do parasitism and suitability for Leucaena. So successfully read on this Irish silkworm eggs for multi-generation. Such ones, improve this performance of the Leucaena on this intermediate host. We also test how about this the Leucaena cultured by Irish silk, the performance control the soybean pod borer. We can see. So just after domestication on Irish silkworm eggs, so the parasitism and the emergence of Leucaena, so the significant overall increasing the trend. The rate of parasitism and emergence from 40% that's in the first generation increased to 90% in 10 generations. So we also could find the performance and the fitness, so gradually increasing. So that's with the generation increasing. So from this, the figure we can find. So on the soybean pod borer eggs for all age, Leucaena come reared from Irish silkworm. So parasite higher the egg reared from rice moth egg. Okay, so so for this the technology, so we also get the patents recently. So another thing important, if we want to expand the technology, so we also need water releaser suitable to the soybean field. We also developed a new type, the ball releaser. I think this one similar to that one, Michelle. So mentioned that one, but this one has different the outside. So some this how to use this this releaser. So this releaser is suitable use utilized with manually and the drone. So finally, see the field extension, demonstration, and application. Yeah, I think because this the trichogramma is an agricultural, so it's important to release this natural enemy. So we should do the good monitoring to the soil import borrow. So we do the monitoring this pest with the sexual pheromone. Also, based on the trichogramma's dispersal distance, we determine so how many the releaser to distribute in the field. So release rate, so per release we release about 100,000 watts per hectare. So total 450,000 watts per hectare. Yeah, so this one for the first year we try this release this parasite so with by hand, but also we try. So later we try how to release this releaser with drone. So this picture for yeah. So in 2019, so we have a field demonstration on using the drone to release trichogramma to control this pest. So another thing I think is important. So how to conduct, to investigate the control efficacy, because this trichogramma is agricultural. So I think it's important we should know the result, for the soybean pod borer, the parasitism in the field. So I think this works similar to collect these resources. Yeah. So we just distribute release the trichogramma in the field. So we distribute how to say that the soybean pod borer acts so from the release center to different direction. So 4 direction. So interview 5 meters, we distribute the soybean pod borer. So from this table we can find. So we can such in 10 meters, reverse in the field, we get the mean parasitism 76. But that one is not easy to do. Just now I mentioned because this the species lay the eggs, the individually. But for us, just do the research we can do. That's investigation, distribute the egg. But for I think the easy measure to investigate the control efficacy. Just prior to the soybean harvest, select local dominant cultivars in demonstration field. So yeah, so where was were released and have the control field. So just before the harvest, we investigate the damage rate to get the control efficacy. Yeah, so now see the demonstration. So in 2018, so we extend to how to start how many? That's 330 hectares. So in 2019, so similar the control the area. So for this technology, so yeah, in 2019, so our CCTV's agricultural channel spotlights this technology. So for the large extension, this technology will also investigate the control efficacy. So we get the average control efficacy 78%. So in 2019, gets the similar to the control efficacy. Yes, since 2023. So this, the products, the technology developed, so have been obtained the public bidding by Jilin province government. So that's since 2018. So we have achieved annual control the coverage of 20,000 hectares for this pest. Yeah, so I mentioned for the soybean, the plantation, the large area in Heilongjiang province. So since 2023, we have tested this technology. So in this province. Also last year, for this year, Heilongjiang province, this technology has a green plant protection demonstration technology for soybean yield enhancement with demonstration and expansion carried out across 1,000 hectares, so province-wide. Yeah, so for this year we do the demonstration, the field demonstration. Yeah, just last month we also have a demonstration meeting in Yi'an County. So the government organizes the field demonstration to have a good doing this the technology. So also we test this the technology, this leukemia to control other caterpillars to damage the soybean. Yeah, we can find. So also leukemia exhibits good performance to this caterpillar. Also for another the caterpillar, so also we get the best performance so from this leukemia. So let's give a simple conclusion. So this technology, so for the control this pest, I think we have had achieved and 7 masters graduate. Also get 5 patents. The achievement. So in future, maybe we can organize, to apply the grants in future. So this technology also realize the transfer to company. So since 2018, so the accumulative area protect through the government support the Trichogramma workforce of soybean pod borer control in Jilin and Heilongjiang province. So has reached 120,000 hectares. So acknowledgement. So we thank the MOST and the MOA and the Jilin government the funding to support this work. Also for this work, so our collaborator, Nico and Wang gave lots of contribution to this work. Okay, that's all.
Okay, thank you, Dr. Zhang. Actually, you know that now the soybean, harvest the surface and the soybean is a pure challenge for China because they're related with the conflict between the China and the US. And I think we imported a lot of soybean from the Brazil. And actually, from MOA, increase in 3 times higher of the fund established for around the soybean seedling and the plant protections. And Lian Shengzang is a specialist professor, only focus on the field application for decades. Actually, all the students here always chasing a high rank publications, but we always think that we should spend every coins we get from the tax by the farmers and pay back feedback from the farmers to get the benefits. So there are some questions around okay. Yeah. Mikhail?
Thank you, Liang Sheng, for the great presentation and the innovation of drone release. Just a curiosity, because we conducted similar research, but with, we'll say, bigger parasitoids. Have you conducted laboratory trials to assess the whole of the sphere, just to be sure about the emergence success? I mean, the parasitoid that comes out. And also, have you tested temperature, the effects of different temperature on the emergency rate of parasitoids inside this carrier?
Oh, so that's a good question. So, you know why we design that different outside the releaser? So we also think about, you know, the temperature. So because, you know, the releaser, you know, distribute in the field, touching the soil, the air. So, but we release this, the releaser. So during, so August, you know, all the soybean, how to that's development stage, you know, have lots of the leaf to cover, you know, cover the seed, the land, the soil. So they can, you know, reduce the temperature effect. So
okay.
Actually,
yeah. So you also maybe mentioned that's a whole.
Yeah, all sides to
yes, that's
let
important illustration. So just for this year, we found another problem. So the ants, yeah, now ants can, you know, pro in this the releaser. So eat, you know, our, you know, parasite, host site. So next year we will, you know, improve the whole. Okay. Yeah, the whole. That's the, you know, the trichogramma can, you know, emergent, but the small ant not enter.
Yes,
yeah.
That's
so you are expert.
yeah, it's a nice point. Thank you.
So Lianshomek is in conservation of reserve all the legs in Delpos status. So he has a very special technique to recover them before release. So whatever the temperature inside of the carrier,
But things are a bit more complicated. See, when you talk about lethality, mortality itself, it's usually straight up relationship directly with the pesticide on the insect, either affecting fundamental physiological process or a symbiote that will be a determinant of the individual physiological process. All good and sound, but we'll focus here much more on other aspects which tend to be neglected. And here we go. I will scan through a few of them. It's not a single study focused on a particular group of insects, it's more of mosaic, scanning through pollinators and so on and so forth. So be patient and we'll get to that. At the start, and the focus here will be significant. And the reason for that is the chances of having prevailing subliminal effects with bioinsecticide is much higher. If you consider raw extracts, you do have several compounds. These compounds, they do synergize or potentiate one another, and frequently enough, especially if you consider non-target species, you are not really looking for mortality, but sublimination. So the part here will be subliminal effects and subliminal responses. I cluster here just a few possibilities. I will not run through all of them, but I will give a glimpse of a few of them. And the first one is this. Those are leaf cutting ants, not fire ants, although it's good preserved. It's a good preserved in Brazil, so China, Italy, and so on. I feel a little bit uncomfortable when we talk about water, because you know where they are imported from. Except they are but there is difficulty. Many of the times, not the target species, but a non-target pest. This is still not target. For anything to get really strong effect on a social insect, mortality is not the ingredient. Because if you are killing foragers, chances are that the nest will not be attacked. Subliminal insects is the key motivator. And in order to have an expressive subliminal effect, you have compromised one of the colony pests, the main colony pests. Usually the queen or the gardeners, and if you're lucky enough, the fungi. So we run quite a bit of studies with leaf cutting nests, and this is a representative one. And the reason is not only the likelihood of non-target effects, but people from forest sciences always look for something to control these insects. So, in one side is a non-target pest species, the other side is a target. And for purposes of forest certification, they are looking for natural compounds, basically compounds that are effective as ant baits against these species. And they both condemned. The usage is maintained while we still do not have replacements. But one replacement of this potential species was a particular compound which was isolated from an eucalyptus clone. They messed up quite a bit with the ant nest. And after a while, we realized that the effect is basically behavioral. If you release the material near the nest, the ants would attack each other to go on cutting legs, heads, and so on. They became extremely aggressive, even with nest mates. And usually in home, the relationship between nest mates are quiet, no problem, no aggression. But not the case here. And on general, you can see that the treated insects, they will fight against each other. Their heads will grow. After a while, were able to identify the effect checking on the critical hydrocarbon profile. And the effect of the compound was exactly on the hydrocarbon profile of the crystal, preventing the nestmates to recognize each other. So fundamental behavior effect. But not enough to allow good control. I mean, the compound was too volatile. Even if you consider alternative preparations. That was kind of interesting study. For those of you who are better acquainted with plant-plant relationship, of course, allopathie is a good indication of this effect that is against plants. And from those plants, you can get quite a strong phytotoxicity. And Michael mentioned that earlier today. I will not waste a whole lot of time. I think it's not really a waste. The hypothesis is insects. So the question here is this showing the possibilities of direct and indirect research relationship. I'm more interested on this. And well, the insect being focus of attention for quite some time. And recently, not that recently, but we saw some interesting cases in which leaf fertilizer was actually causing quite a bit of harm on these insects. No, it's a non-target effect, it's not even a pesticide. But depending on the leaf fertilizer you use, in the salt ones, particularly contain very particular to the insects. They compromise with but they do it as well. Is substitution, the human treatment, toxic compound actually is preferable and leading to a series of non-target effects. Again, still considering stimulus, people tend to believe that honeybees are a good model for this particular group of tropical bees. And that's not really the case. You'll be seeing completely opposite effects when comparing stingers with honey. That's just one example here with the spinosad. Again, a natural compound, a product of fermentation. That you can see it compromises longevity, it compromises weight gain provided by the lab. And with that, it compromised development, leading to adult malformation. So the insects are actually able to live in adults who reproduce, but malformed and with learning difficulties, appropriate problems. Okay? And a further ingredient, we tend to think of insects as an isolated individual. And insects actually house quite a bit of products, and a number of them are essential. So we do have a curious case here with the mice weevil. And historical products, they don't have a very diverse symbiont form, but these weevils have at least two symbionts in one. A primary in the symbiont and both backward. If you do knock out somehow one of those, consequences will take place, particularly with this one. If you under heat treatment or using any kind of bactericide or antimicrobial, you do compromise food consumption, respiration rate. You will slow down weight gain, leading to behavioral difficulties and eventually impairing fertility. So it's a cascade of effects that will have population effect. But so far, we've been just exemplifying a few of the potential effects, considering just the order. So, comparing physiological processes within the insect. I will change a little bit to focus on potential effects on populations. And not, again, a series of very long series of cases, but I will focus on one particular phenomenon that I've been working in for which is psoriasis. which sometimes is weird to explain, but the concept itself is simple. So pay attention. You probably saw already this somewhere. I don't speak German, so don't worry. But I do have a translation in here. If you are not much of the philosophical person, so hold on, I have a solution. And this will do as well. So I'm plain English. But that's the notion. Yeah? What doesn't kill you makes you stronger. Well, most of the time, it's not always good. Keep that in mind. Now I will show this as a plot. But remember, we were talking about little effects, not little. Little effect just kills you. That's it. If you want to know, if you want to learn if there are any substances due to biological activity, what you do, you exposing population samples to increasing concentrations of any given compound and check on the response. What you do expect is an increasing response with increasing concentration. Not mortality. So if you consider this as reproduction, well, zero high reproduction, increased dosage in reproduction compromises. Okay? If it's mortality, everything goes up. But should we do that? You have something like this. But if you do have organisms, well, the trend will be you do have a peak before the inhibitory phase of the relationship. So it's a biphasic concentration response. The regular pattern is monophasic, just a single frame downwards. Here you have up and down, so two different phases. Looks weird, but remember, if you consider heavy metals, for instance, heavy metals are quite toxic to humans, but they are also essential for a series of enzymes. This is what we talk. So in trace amounts, they are actually essential. If the concentration in the body suppresses a threshold, bad consequences will take place. If you need a more gentle example, consider alcohol. A little bit of alcohol. Well, some say it makes good, particularly wine. But keep in mind, that's the trend that we are looking for. The question is how frequent this phenomenon takes place. We don't know how frequent, but we do believe that it's much more frequent than one realizes. And here are just three examples, and I'll focus on the fourth one. I think bug predator, for increasing a little bit concentration of a pyrethroid to actually favor reproduction. So population growth goes up. Next comes being beetle exposure to azadirachtin. And texture. Again, the truth that insects have better reproductive performance under a limited range of concentrations. And even in plants, and we did a few studies with coffee, soybean, pineapple, there were some systemic insecticides. You can actually see an upward trends on intermediate doses. And sometimes, or actually quite frequent, they tend to match the concentration used on seed treatments. And in Brazil, they joke around that this some time ago that when the coffee price was quite high, they used pesticide as fertilizer. Because they will use alcohol. And very early on, because it will boost vegetative growth. And the coffee beans, they will come up on new branch, so the growth is up. But nowadays, not that frequent anymore. I would say something is much higher. A probably more interesting case is this. 2001 is the second year past and maize cotton in Brazil. Amazing cotton in Brazil. We do have toxins expressive. And if you check, this is a lab experiment in which the the BT toxin was provided in the diet, and you can see the development of the life was quite good. We had relatively lower dosages, considering the range used on such treatment, which is unusual, but it's a good example of releases. Yes. And this range actually matches the range expressed in the plant. So you do have positive consequences for the insect. The insect is better on the plants. You probably heard a lot that one of the justifications of usage of BT, maize, cotton, and soy was the potential reduction of pesticide usage. Well, on paper looks good. In reality, at least in Brazil, that didn't take place. 10 years ago, the average usage of insecticide in Brazil was in the range of 12 kilograms per hectare. Today is over 20 on average. So despite of quite large pattern of BT used in crop plants, insecticide actually is quite small during the same time period. So there was no reduction. And if you consider secondary pests, they usually are not affected or they might be favored by the toxin at the period of the particular flower. If you consider that the primary target is already resistance, you have a double crop. It is not really working. And with that, pesticide will increase, of course. Okay, so far we checked what took place inside the insect, and then on the population outside the insect. I will try to illustrate what might take place when you do have interactions. And again, here, there is an old thought in the field, pyrethroids and pyrethrin would favor mite outbreaks, particularly red broad mite. Because they have differential susceptibility when you consider mite pests. And the main feature is the predatory mite, but that's not really the truth. Actually, the insecticide was quite toxic with the pest, both of them. Selectivity was pretty close, but you do have releases of the red broad mite. So lower range of concentration will actually favor the outbreak of the red blood. That was the key problem. And as things tend to grow even more complicated, insects do not live alone. This field applications of the insecticide, chances are that you're going to be affecting several. So you need to figure out not only the effect on the main pest, but all the others that will be coexisting and interacting with that. And a lot can take place. Again, just an illustration of releases, and if you consider community-wide effects, so if the control is that, you might end up with a simplification of the crop network, or even an enhanced complexity if releases takes place. The problem is to check this is difficult. We tried some things. First on with bottom. And well, this is just a spatial representation of the different treatments, BT and non-BT, and 2 seasons of cultivation. And all of the treatments are very different. So difficult to get out of that. But in the end, we just realized that the largest difference regarding crop network was among season or year, not much on insect. But this was a short period assessment. What 1 point, which not 10 years. Eventually, the cumulative effect much higher when BT is under usage. Well, we checked that in cotton, but we checked in maize, and the result is basically the same. In maize, we went even further because we tried to figure the spillover effect. So in Brazil, we recommend that 20% of the areas should be maintained with no crop. We are curious to know if the non-BT will be interfering with the insect community on the BT crop. But yeah, not much effect. The main difference are really the season. There are exceptions. When a cumulus one is represented as here, then we tried seed treatments on soil. We observed that the assemblage of total richness and abundance of insects, they did not change much. But when we consider the community interacting species, we actually realized that in some cases, with seed treatment, the food web was more complex. Trust network was more intricate. And this was very clear on the second. There was not really great change on yield, but there was differences on the profit. And again, it reflecting the possibility of community-wide organisms in such a case. Just a speculation, not a whole lot of data, and the short-term effect, not long. So to close it up, keep in mind that bioinsecticides are still insecticides. Okay? They changed molecules, but the problems are still there, and you need to watch out. Especially if you consider that when we talk about bioinsecticide, you're talking about a lot of stuff clustered together, and they're not the same thing. Sources are completely different, toxicities are completely different, the structures are different. The fact that it's a bio compound doesn't mean much, since the structure is what determines the physical chemical properties of the compound, and those properties are the determining safety. So the molecule tells you about, and it's a case by case. So no generalizations here. Okay. So yeah, you can change molecule, but usually you still think the same way. Why you probably should. I'm not particularly optimistic, and my students tend to complain. But I thought of maybe something inspiring. Have you ever heard of this triangle of life? So there it is. Just choose two out of the three and see if you can. But yeah, that's basically what I speak like to provide a few examples. Just a note of caution on dealing with this sort of problems. With that, I think I can apologize. No time for question, I think. No problem.
Okay, thank you very much. I think if you have some questions, then you can communicate. Sure. Yes, later. Okay, the next speaker. So because we still have time this afternoon and tomorrow for field visit, so if you have any questions, you can continue for exchanging the idea. Okay. The next speaker is Dr. Mary Sher, I think, from the University Polytechnician Valencia from Spain. Yes, for me it's a little hard. Okay. And I think she will present the work on plant-to-plant communication for SK2. Okay, welcome.
Thank you. So, good afternoon, everyone. First of all, thank you to the organizers to invite me to share part of our results with all of you. So to start, I would like to do a brief introduction about what is that of plant defense mechanisms. In nature, plants are in continuous interaction with the ecosystem. The occurrence of abiotic stresses like drought, flooring, salinity enhance plants to develop several mechanisms to turn off this disturbance, involving the activation of different biochemical pathways leading to the production of either defensive compounds or morphological changes. In addition, plants also activate defenses against a plethora of attackers, including the herbivorous arthropods and the plant pathogens, where the insects are the more important invaders. The response that elicitate the plants can be divided in two categories: the constitutive defenses, which are present in the plant before the insect attack, and the induced defenses that which are I cannot see the screen there, which are produced only upon the constitutive defenses. Can be either physical and chemical barriers against the insects, such as trichomes, spines, rust. And the induced defenses can be divided again in two categories: the direct defenses and the indirect defenses. The direct defenses includes the compounds like protein inhibitors, polyphenols, peptides, compounds that could be anti-nutritive or toxic for a lot of herbivores. In addition, after insect feeding, the combination of plant damage with the elicitors from the herbivore induce the production of secondary metabolites for the plant, which result in the emission of specific volatile organic compounds that are called herbivore-induced plant volatiles, which result in attraction of parasitoids. And it is called indirect defenses since recruit natural enemy to combat. So, in short, through herbivore-induced plant volatiles, plants communicate with each other by emitting and receiving volatiles, and these plants that receive the warning volatiles enter in a state of defensive activation to anticipate the arrival of the aggressor. What is clear is that plants cry for help. Well, thank you very much. So, on the other hand, I would like to introduce you the Phytophagous predators. As most of you know, during the last decade, biological control programs in greenhouse tomato and other crops has been successfully implemented using these Phytophagous plant bats like millets or arthropods. In general, they can feed on bones, plant tissue, and in a wide range of foods. So they are really good predators, but also cause phytophagy on the plants. For more than 10 years, we could demonstrate that the phytophagous predators do to the phytophagous behavior active the same defense mechanisms that trigger herbivores. In our previous trials, we realized that in tomato plants with presence of Nesidiophorus tenuis, one of these Phytophagous predators that we use in Spain, these tomato plants had a low incidence of white fly. Our first thought was that it could be due to a direct effect through predation. But later, we started thinking that it could be due to a indirect effect through volatiles that emit the plants that can cause the repellence of the wildlife. So this is it. It was our first model, and we could verify that the phytophagy of the predator, Nesidiophorus tenuis, activates the metabolic pathway of abscisic and jasmonate acid in tomato plants. We made these plants less attractive to the whitefly and more attractive to the whitefly parasitoid environment. We confirm that tomato plants previously activated with this Tophophilus predator, the Sidiophorus temis, have a lower infestation of Tetranicus fungidae, the two spore spike mites, due to the increase of different proteins in the liver, as you can see here. On the right. Here you can see that 14 days after infestation with Tetranicus fungidae, we had lower populations, 35% lower of females for plants, in these plants activated with the Sidiophorus. In another bioassay, we confirm that also the phytophagy of the predator, the Sidiophorus temis, induced the early expression on glandular sticones, number 4, and of course the production of acyl sugars that produce this kind of So here on the left, you can see after regulation in some markers of the jasmonate acid pathway with an increase of more than 60% in the number of trichomes in these plants activated with the Tophophilus predator Sidiophorus. Of course, after to increase these trichomes, we increase the production of acid sugar by these plants. So in the last example of induction of defense by Tophophilus predators, we confirm that the defenses activation mediated by Tophophilus predators managed to limit the accumulation of the tomato spot wild. So for in this assay, we use two Tophophilus predators, Macrolophus pygmaeus and Sidiophorus temis, both of them activating the similar metabolic pathway of jasmonic and salicylic. The more important thing was to see that both predators reduced the viral cycle 21 days post-infestation. And here also, picture is worth 1,000 words, so you can see the control plants with the typical symptoms, with the spots on the leaf, with those symptoms in the plants activated by both with the both So taking again our previous model, we observe that volatiles emitted by the plant culture by the Sidiophorus temis induce defenses in intact neighboring plants by activating the jasmonate acid So these plants that never had been in contact with the Sidiophorus or another Tophophilus predator just were in contact with the volatiles of the activated plants were more attractive to the parasitoid of whitefly and Garcia. And these results could be a reasonable explanation of the great success of the Tophophilus predators in agricultural crops. At this point, we investigate which of these volatiles are responsible of this activation of defense in plants, with the goal to choose one of them for a new control method against pests and diseases based in communication. For that, we identify all the profile of volatiles emitted by these plants, and we select some of them, like for a responsible of activated defense in tomato plants. So for that, we expose individual tomato plants to each of these medically volatiles at the beginning for 24 hours, and also at the beginning with a very basic emiso, a piece of paper with a very low concentration of these volatiles. And we analyze different marker genes of the jasmonate and salicylic part. So after this analyze, we just move forward in this research line with two volatiles, the hexene propanoate and the methyl salicylate for all the bibliography. So also we studied everything in response of 4 tomato pipets and 1 parasitoid using a white tube olfactometer that consists in a glass white tube connected to 2 jars where we put inside the test odor. These jars connected also to an air pump. And we studied for the behavior of 4 pipettes, Frankinella, the thief, Benicia tabasi, the whitefly, Tetranicus urticae, and the Microlepidopter tucapsoluta, also the parasitoid. So you can see, using both volatiles, that 3 pipettes, the thief, the whitefly, and the Microlepidopter, prefer the control plants than the plants that have been activated by exposition with the volatile. In contrary, the parasitoid and Tarsia formosa prefer the plants that had been exposed to the volatile. Also, we study the clasticon and metabolome of these plants. I'm not going to enter in detail of this USA, but in this heat map, you can see these 2 columns are columns that corresponds to the activated plants. The transcripts are totally different than the transcripts that we found in that plant. And it confirmed that the genes involved in specialized defenses were abnormal. Also, we wanted to know if exposure to these 2 volatiles could reduce the subsequent herbivore infestation. For that, we use 3 treatments, the control plants just exposed for 24 hours with the volatiles, or plants exposed during all the PSA. So we measure the number of eggs laid by Tucapsoluta and the mortality of the microlepidopter. No difference were found when we used in both the number of eggs and mortality. No difference were found when we just activate the plants for 24 hours. But when we use a permanent exposition in red, we could reduce over 67% the number of eggs per plant, and also we increase the mortality to more of the 80%. No difference we saw in using methyl salicylate, and this was the reason to just follow, to move forward just with hexene. So at this point, also, we wanted to arrive to a large areas. For that, we need a dispenser. And thanks to a collaboration with a group of the Polytechnic University in Valencia, you could identify and select one specific dispenser that we can live in the field for more than 4 months with an emission constant and a slow emission about 10 micrograms a day when the temperature is around 25 degrees. And thanks to the dispenser, we can move to large area. This is resource in a semi-field conditions. And we can see that during 2 months, we could keep activated the jasmonate and salicylic pathway in the plants activated with the hexene propanoate. More surprisingly, it was with non-preneurs. So these plants are the same height. So the plants, the control plants and the plants activated with the volatiles have the same height, and more important, the same food. Also, the last step was to jump to a commercial. So before to hang the dispenser, you can see in the different greenhouses that in the expression of jasmonate and salicylic marker pathway, you can see the same level. But once we hang the dispenser in the field, we 4 and 8 weeks later, we keep also with all the interactions that you have in a commercial field, the workers, the harvest, etc. So are activated this factor. Also, more important was to see that the population of fruta absoluta in these houses was lower in the houses where the emissor was high, but with not disturb the population of our Tropicophagus predator, the Syriaconis. So for this first part, just I would like to say that our work demonstrates for the first time under commercial greenhouse conditions how the exposition of volatiles in use of plant defenses through polymeric dispensers can be successfully integrated as a new biorational and sustainable practice. And thanks to these exciting results that we found in tomato, we start to thinking if we could induce also other crops with these volatiles. And we wanted to answer this research question. The first was if we could, through exposition with the volatile, induce plant defense in sweet pepper. Also, if we could manage different species in sweet pepper, Spodoptera, Fentimella occidentalis, and Trialinodes. Also, see could affect the development of Nesidiocoris stenis in sweet pepper. And at the end, as it was a new question, if we could enhance plants' tolerance to water. So to answer this objective, for the management of the tricky pest, we conduct 3 separate experiments, one for each species, one for the fly, for the trip, and for Spodoptera. We just activate the plant for 2 days, and the day 0 we infest with one of these pests for each USA, and we assist weekly different number of adults, larvae, exclusion media. Also, experimental design we did for to know the development of the things. And to know if the exposition of the volatiles would induce the plant defense in sweet pepper through transcriptomic analysis, we conduct a we did the extraction of RNA, and we performed a RNA assay to analyze the gene difference profile in these 2 kinds of plants, the control plant, the effector, and the plants activated. Also, for the last objective, we analyzed, we performed a gas exchange analysis in a climatic change under controlled conditions, and we measured the different physiological parameters 7 days post-activated with volatile. We measure the rate, the stomatal contents, the water. So for Spodoptera exigua, you can see in the figure how the number of Spodoptera exigua was 92% lower in these plants activated with the volatile in comparison with the control plants. The same level, also the consumer area was reduced more than 90% in these plants activated with the volatile. Again, the pictures, you can see a destroyed plant in control plant with yes damage, but much less damage in this one. For the tree population and damage, also we could reduce the population and the damage in these plants activated with the volatile. And for the whitefly, also we could reduce the infestation and was significant after the to study the effect on the development of Merciliopolis berries, to say that we didn't see any negative effect. In contrary, so we could enhance the establishment of Merciliopolis berries after 20 years. So about the analysis of the gas change, to say that no difference we saw in the photosynthesis rate. However, when we measure the stomatal concentrations in these plants activated with the volatiles, was a lot reduced, and also it good to enhance the biotic stress, because to close the conductance, you restrict the entrance of pathogens inside the plant. Also, in the same plants, we could increase the water use efficiency. And it's good because the plant lost less water by transpiration. So, and it could be good in water stress or drought. So also to see if this volatile could induce plant defenses with pepper, we did a really asset analysis. We identified more than 20,000 genes, which of them more than 1,200 were differentially expressed. Almost all of them upregulated. To say very briefly that 62 genes were involved in activation of antioxidative machinery, and it may stimulate oxidative stress and thereby enhancing plant stress tolerance. Also, these plants activated with the volatiles. We found 40 genes involved in the activation of defense relative to wounding and pathogenesis, and plays a pivotal role in activated plant defenses against herbivores and diseases. Just to finish, say several conclusions that the volatile exposure in sweet pepper reduced the infestation and damage of Tranquinella, Spodoptera, and the whitefly. That these plants enhance the establishment of the biological control agents like Merciliopolis berries. These plants reduce the stomatal contents, and it could protect against pathogens. Also improve water use efficiency, maintain plant productivity under water stress, and of course, induce a wide array of plant defensive response, enhancing the stress. Yes, key to take away that use of volatiles as plant defense elicitor is a sustainable pest management tool to enhance biological control strategies, which can play a role in the management of herbivore pests in sweet pepper and other crops. We will see later also in citrus. And to enhance plant tolerance to water deficit change. So just to say thank you to all the collaborators that are behind this work, and especially Alberto Urbaneja, that is co-author of almost all these work. Of course, thank you for your attention.
I have a question. Just want to know. So this the compound you found, whether attract or drive the net enemy.
How you, because change the profile of the volatile that emit the plant.
the volatile, some volatile to can attract parasite or predator. So I want to know how about this volatile, how this function.
What is the function of the volatile? So the volatile activate the plant, so and change the profile of volatiles that emit the plant. A lot of them related with the jasmonic acid pathway.
Yeah, so that's my suggestion. Maybe you can test in the future, because this volatile can enhance the defense, right? The plant defense. But how about the interaction with natural animals?
Okay. So we have another bioassays also, and we know that just not through the plant. So just the volatile is able to attract natural enemies, predators, and parasitoids.
So
Attract, yes. Attract. Because it's like a volatile that the plant emits when it have some kind of damage. So the plant cry for help, like I told you before. And this is one volatile.
Most volatile induced by how the function to attract the phosphorus. So, but this we found a new compound. So maybe also have this function to attract.
Another compound. We use a lot of them. Yes, they can work. But for us, the best is hexavyl propanoate. But also there's another esters, hexavyl butanoate. I don't know. Is this the question? Next or no?
Did you test this compound from that one? How the function to attract the parasite? That's my question.
The mechanism? The mechanism? No, the mechanism, I don't know really the mechanism.
So, yeah, you have found that's the volatile, right? And now become the product, right? So yes. I want to attract the Acacia formosa or drive Acacia formosa. Attract.
So, these plants, when are activated, repel pests and attract natural enemies. Our model was Acacia formosa, but also we found in the open field a lot of parasitoids. So, if you put the volatile with a yellow sticky trap, so in 1 week you have the sticky trap full of parasitoids. It's amazing.
So, because you also test this defense to caterpillar, so on the pepper, right? So that's the second experiment. So I also want to know how about attract the other natural animals, so that's attack the caterpillar. So that's okay.
Yeah, yes. Okay, nice. Okay, the nice presentation and the nice question. Okay. Okay, next, welcome Dr. Alberto. Okay, nice. Also from Malaysia, Spain. Okay. And I think you will introduce the work on the multiplication of IPM strategies. Nice. Welcome.
Thank you very much for the invitation and thank you for your attendance today. I'm going to take about citrus, building on conservation biological control, and multiplication of IPM for resilience in citrus. Well, this is the line of talk. The word is really short, just 30 minutes. And before, I would like to introduce a little bit why we are really interested in citrus. Citrus in Spain is more than 300,000 hectares of citrus. We are the main producers of citrus in Europe, the first exporter of citrus in the world, and our pest management is based in conservation biological technology. We have a very robust system of biological control. We have been introducing natural enemies from the beginning of last century, 20th century. Apart of this, the main characteristic of our citrus now is that we are free of very threatening, very dangerous diseases like the citrus one that this morning already was introduced. We are free of HIV, and this allows us to continue with conservation biological control. Low input of pesticides, you will see, and we are trying to protect and to enhance the efficacy of natural. I'm located in Valencia, in these areas of central west of Spain, where almost 200,000 hectares of citrus is produced. And my institute, the Valencian Institute of Agricultural Research, is in the middle of the citrus. So we are a center focused, I would say 70% of our research is devoted to citrus research. Our citrus are attacked for more than 140 hectares, but only some of them can reach the category of pest. Why? Because we have a very complex group of natural enemies that maintain this type of pest below the economical threshold. As you can imagine, our citrus industry at the moment, comparing with other regions in the world, we can say that our citrus production is relatively healthy in comparison with other citrus areas where they treat our against the vectors of these diseases. However, this idyllic scenario is changing from the last years for different reasons. The first obviously is the climate change. Many dynamics of pests, also natural enemies, have changed. Some pests that in the past had 3 generations per year, now we have 4 generations. And this 4th generation is producing damages, almost what we consider winter in the past, and now is autumn. Okay, so this is one of the first problems that we have, and the second main problem is the appearance of zoonotic pests. Especially in citrus, we are welcoming almost 1 pest per year. So this emerging threat, as you can see in this graph, in 2020, you can see that almost 1 pest is appearing in citrus. Some of them appears causing minor problems, but some of them are producing really serious problems, like the South African millibug, the Notococcus aviaris, or during the last year, the thrips problems, especially earth-inflow thrips around. This problem, this is the pests that are appearing in citrus, but we are very close to the HIV. Okay? The Mediterranean region is free of HIV, but the vectors are already present very close to the Mediterranean area. The African citrus psyllid, Triotha erythrae, is already present in Spain, but not in the citrus area production. It's in the north of Spain, which is strange, but we have some isolated citrus in the north part of Spain where there are no production of citrus, but Triotha is present there. And the Asian citrus psyllid, Diaphornia citri, is already present in Cyprus, the first detection in Europe, and also in Israel. Okay, so all this situation probably will change in the near future. So we are, in our institute, in collaboration with other partners, we are approaching these problems, invasive and also future problems, with a dual research approach. One is that we are first targeting the pests that appear in our system, targeting new specific programs to try to reduce as soon as possible the population levels, because we know that this is historically always occurring. That when a pest appears, growers start treating, and this disrupts our system. So from the beginning, we know this, and we start working very hard to select the most selective pesticide, because at the beginning, always growers have to treat. Okay, and of course trying to find other alternative solutions, establishing the thresholds, looking for natural enemies, etc., etc. And the second approach, that this is for future programs, we are trying to incorporate multifunctional and holistic IPM program, basically to enhance the resilience of citrus, to make our citrus more robust, to be ready to future programs. Okay, and one of the main targets that we are working very hard in the last year, obviously, are the vectors of HLD and HLD. That is our main three. Well, which is the different techniques or programs that we are looking for. I'm going to enter in one of its circle, okay? Biological control, both conservation and classical biological control, functional biodiversity, plant defenses that already introduced by Che, nanoformulations, and also tolerance to antibiotic stress. The idea is to incorporate these systems to make the citrus more resilient to this pest, and also don't forget that this is very important for us, is any new tool that's been incorporated to our IPM program, based on biological conservation, biological control. Because we know from many years ago that this is the basis for our citrus, and we have to maintain our pest always with natural enemies. So I'm going to give you some examples of our integrating tools that we are using. First, classical biological control. In the last 10 years or less, we have launched two different classical biological control programs. One is with this parasitoid, Anagyrus atriae. It's a South African parasitoid that we imported from South Africa to the South African mealybug. Okay? Very successful, especially during the last 3 seasons. The population dynamics of South African mealybug now are decreasing, so it's producing the very good new results. But every year we continue releasing the natural enemy in order to regulate the population. And the second example, the Anorexia 3, I'm going to enter a little bit more in detail, and another one that was not introduced in Spain, but we forced to introduce this parasitoid, Anorexia radiata, to citrus. I will also explain these cases. In 2018, 2 years before we moved to South Africa, we prospect different parasitoids. We selected this one, the Anorexia 3, as the most efficient in South Africa, and also after conducting all the legal requirements, specific and accomplishing all the procedures that our administration requires. This morning, Nicolas explained that it's not easy to introduce a parasitoid. We got the permission to introduce the parasitoid in the island, and 2 years later, after the results in the island were amazing, we obtained the permission to release in the north of Spain. This is what I told you. This isolated citrus, this is not commercial citrus. We release the parasitoid there, but the main citrus production is in the coast. Okay, so look, this is a good summary of what we are doing. This is in North Spain, 3 different provinces. In the first province that we released in Pontevedra, you can see that after just the first release, this is the percentage of fields where the Triotha erythrae, the South African mealybug, is present. You can see that in just 1 year and a half, the population level were reduced at all. The same in the following years, okay, and the program was very successful. More interesting is in the Atlantic coast of Portugal. This is the areas that were infested by Triotha erythrae. We were at one point that we were very worried because this is very close to the citrus production in Spain. That is this part of the program. So when Triotha erythrae was detected in South Portugal, we really were worried. But thanks to the releases that we conducted in the north of Spain and also in Portugal in collaboration with the Portuguese government, in 2025, it was declared Well, this is in Portuguese. Maybe our colleagues from Portugal can understand, okay? But what is important, translating is we could eradicate, this is the word that the Portuguese use, we could eradicate the Triotheriferae from the south of Portugal, okay? I know from this last year, in Galicia it's also very hard to find the Triotheriferae, the African citrus lily, and in Canary Islands, the first place where we released, to find Triotheriferae is also very, very difficult. So this was a very successful program of classical biological control that allowed to maintain our Mediterranean citrus still without this important vector. Regarding to the Asian citrus lily, I told you that it was detected in Cyprus. So we started a project in collaboration with the Cyprus government and with the University of California, and we introduced Amorixia radiata, a strain that was selected specifically for California, a different strain that is used in Portugal, in Brazil, and is used in Florida. Okay, it was a strain that was collected from Pakistan, and the parasite was released in May last year. Okay, in 4 different points, and at the beginning of this year, we visited citrus again, and the parasite was already established in the island. Okay, the levels of Diaphorina citri were reduced. Okay, we have to continue confirming this. Okay, so this is also very successful results that have to be confirmed now in autumn. But more interestingly, and this is for the first time for this Asian citrus vector, we found another native parasite. We found this parasite, Amorixia citricola, that was also present in very high numbers in the island, parasitizing Diaphorina citri. Okay. Well, this is very interesting because we know that this parasite is native from the European, from the Mediterranean basin. It's not only present in Cyprus. We know that is already present in Greece, in the continental part of it's already in Europe. So this is interesting for the future human populations of Diaphorina citri. So now we are really, really interested on this new parasite of Diaphorina citri, Amorixia citricola. Another approach, cover crops of functional diversity, another example that I'm going to explain very briefly. One of the problems that we have in citrus are aphids. There are two main species, Aphis spiricola, the citrus aphid, and Aphis gossypi. For us, it's much important Aphis spiricola. Both aphids have a rich complex of natural enemies, but okay, this is really important for us because it's producing these symptoms in the new flushing. You roll the leaves, so this can produce a reduction in yield. So, especially in mandarin clementines, we have to do Aphis spiricola. So for that, as I told you, there is a rich complex of natural enemies, hoverflies, coccinellids, parasitoids, etc., etc. So we wanted to increase the natural enemies using cover crops, right? So at the beginning, we selected this cover crop, Festuca luminacea, right? It's like a cereal, and we wanted to test something like pancake plant. We released there some cereal aphids to release natural enemies, and then to produce the factory in the cover crop to reach the citrus. So all this was not necessary because as soon as we planted the cereals, the Festuca luminacea immediately was infested by native aphids, Cytodium avenae, some cereal aphids specific from cereals and not for the dicotyledons like citrus. And this cover crop immediately was colonized by natural enemies. So indeed, if you just in some details, the red one is the Aphis spiricola, and the rest are different species of aphids. You can see from the beginning of February, the cover crop was colonized by aphids, okay? And until the end, high population of Aphis spiricola arrive. So with this, you can see that this is a study that we conducted some years ago, different orchards. Each orchard is one replicate. We conducted 8 orchards with cover crop and 8 orchards without cover crop, very short. And you can see that the threshold was not reached in those orchards with cover crops. Okay, so this was very interesting results. So at that moment, beginning of May, the flushing are so hard, so it's not needed to continue because already the production is on the yield is already. So from then, I'm talking about 4 years ago, cover crops every year is more common to be used, especially these areas. But not only this, we're also working in functional biodiversity, increasing or improving the cover crops, adding flowers as resources for nectar and pollen. Okay, and also we tested this in different crops, and we obtained, for example, that the efficacy of the cover crops depends on the area. Okay? It's not the same to do in Valencia that is separated 400 kilometers. And also very important, the cultural practices and how you manage this cover crop. For example, if we mow the cover crop 3 times per year, there are no effects, okay, in comparison with solar array. But we only mow the cover crop once a year, we can increase for example, degradation on the tree, okay, on the canopy of the tree. We use some sentinel brace to test. So this is also very, very interesting. Indeed, in citrus, from some years ago, we are also a part of conservation biological control. There are companies that are starting to produce and to sell natural enemies specific for citrus. This is the 3 species that are most commercial available now: Phytolemus monticerii, controlling bugs, and Anthilus vladimiri, and Eusebius californicus, that is widely used against spider mites in citrus with this kind of shrinkage. Okay, so this is also increasing in the market. At the moment, you can see that there are almost 40,000 hectares where augmentative biological control is conducted in citrus. And conservation, as I told you, is very important for us to maintain the natural enemies, and we have a good system from 40 years ago that any pesticide that is going to be used in citrus first has to be evaluated to know the side effects between the 3 groups of natural enemies: coccinellids, then the coccinellid predators, maybe the cardinalis, parasitoids, key natural enemies, parasitoids like Gades moaki, and predatoids. Also, stipulatus is very key for us. Okay, so this is our web page, IPM page. You can check easily if any pesticide, also an APP to test and check all the pesticides. Okay? Another strategy that we are dealing with is the planting different solicitors, the HPVs. Mericheria already introduced the approach, okay, is to hold 1 dispenser per tree that we are using, and we make more robust the citrus tree. We have very interesting results. Okay, this is the transcriptomics on citrus. We are able, just exposing this volatile, to increase the antioxidants in citrus, to increase the defenses like pyrrhic defenses, resistance to pathogens, to activate different pathways, mainly salicylic and japonic pathway, and also to produce some metabolites that are responsible to repel and to attract parasites. Okay? And also, well, also we have demonstrated that we are able to downregulate the PAP21, this molecule that this morning was presented. Okay, with the activation of these defenses, we know that we are also to regulate the balance peak. This gene really important for the basic pump. Anyway, we know also that many genes are upregulated related to secondary metabolites, so we also to test in the field if we can obtain good results. And this is a 3-year study. When we started holding 1 dispenser per tree, this is a commercial orchard, okay? Very big. When we designed the experimental block design, and we evaluated during the 3 years different population levels. Today, just I showed you that 60% of reduction in aphid infestation, thanks to the activation of plant defenses. Okay, you can see the 3 different years. Other approach that we are using is nanofertilization. This morning, Michele Recupero also explained the importance to nanovehiculize the plant extracts, okay, in order to avoid phytotoxicity. So we have been playing with different citrus pests, with plant extracts, and for example, with garlic, we have designed a bait treatment with garlic, nano garlic, and we can attract the lead fly. The lead fly is killed. Okay, so this is very interesting, and also we have very good results with the South African bait. And more importantly, we were able, using some of these plant extracts, to activate the plant defenses of citrus. Okay, so this is also a double positive effect of using these nanoparticles. Okay? So this is a approach that we are also to integrate in our system. So just to summarize, okay, we are trying to improve our APM system that is based everything in conservation biological control, trying to incorporate tools that are addressed or target not only for one pest, as you can see, but there are other positive effects. Cover crops, I showed you that is a positive effect on aphids, but are also positive to other pests. Okay, also to increase some characteristic of the soil to avoid erosion, etc. So each of these strategies has multiple benefits. So this is the idea. So finally, our goal is to maintain with a healthy citrus production in Mediterranean basin, okay? And obviously we are also open to new technology like gene editing, natural molecules, small molecules, precision sensing, etc. And of course, with international collaboration, this is much easier than doing this alone, okay? So from our idea is always to be open to collaborate, because the benefit that we are looking for is not only our benefit, it's the benefit of the agriculture and the okay. And finally, Maricel, author of all the articles, and our colleague, and director, Alejandro Pena, also participated in some of these works. Okay, so this is the funding that we have received, and thank you very much for the attention. Any question? This is important. We have a digital repository, digital place. Maybe here, you can find not only the publications, also talks and outreach publications and transfer and all this kind of things. Okay, we are obligated to all these publications there. So now, thank you.
I have a question. Yeah, I was wondering, when you mentioned what one pest was eradicated, so do you think really it's eradicated?
Good question. I say the Portuguese government say that it was eradicated.
But it was not. Yeah, no, it was still important.
Yeah, but they say that was eradicated from this area. This is the word that they use, eradicated. You can check the word Portuguese.
What do you think?
Yeah, I think that it's not easy to find it. Indeed, some colleagues were there prospecting, they didn't find it. And some years before, we obtained the same in Canary Islands, but our government didn't say that was eradicated. No, that's different. Probably it's still there, but what is clear is that the population levels are very low, almost zero. This is a funny history about that. We were in a project, national project, collaborating with different regions of Spain, working on Triotheriplen, African citrus, and we released from Valencia. We released the parasitoids in Canary Islands. And other colleagues in other parts of the dynamic populations, they were complaining us because we finished with the population before the project finished. So it was really difficult.
Well, I was asking this question because it's a very, very good example of achievable classical biological control. So the two populations, the best, naturally, they are there, but super low density, so you cannot find that they are still there. And many people when they ask us about what happens when the target pest will disappear, what will happen for the natural environment. Actually, the pest will not disappear on the natural environment. So just very low density. Very nice.
Very amazing work. So I'm quite interested in the work you have done that you released parasitoids to control the mealybugs. So my question is very simple. I'm very curious about how did you do the very big release? I mean, you released it across California. So, yeah. So it's like a very big work.
Yeah. Thanks for the question. Indeed, these are two examples of classical biological control, two totally different. In one, the one that we were talking now, that the population were almost eradicated, we released very few amount of parasites, probably 300. And with these 300 parasites at the beginning, totally control the area. So we only conducted 2 releases in Canada, for example. Okay? But with the one that you asked, the juice, we are producing, I don't remember the numbers, but probably we already released around 1.5 million parasitoids, and weekly we release all the Valencian citrus area. We have different release points that weekly our technicians go there and release in one orchard 1,000, another orchard 2,000. So it's a release that are continuously being supported by releases. Because the efficacy of this parasitoid is by far different of the another parasitoid that was included. Okay? So our idea is that at the end, all these parasitoids that we are releasing are able to regulate, and we already obtain naturalized population, but we know that the parasitoid is established, but we are reinforcing the releases. We release, because we have a huge factor with this parasitoid, we release almost 3 minutes citrus area of the Valencian community. We have also exported this parasitoid to other regions, so it's different. It depends on the efficacy of the parasitoid.
Okay, nice. The first session we finished is 3 presentations. The topic on bio insecticide, the non-targeted bio insecticide, and the volatile induced plant defenses, and also multi-functional of IPM strategies. Very nice talk, very nice presentation. Thanks again. And then now we have a short break. Then we come back at 3:40 p.m. And then the next session, my colleague Dr. Shaw will organize it. Okay, thank you.
in insect toxicology and pest management, with extensive experience in developing eco-friendly control strategy. So let's give a warm welcome to Professor Heidi. You have 30 minutes.
Yeah. Try to be short. So good afternoon. Thank you for the presence, and I'm really, really happy to be back once again here. It's an opportunity to meet some friends. Some of them they have been seen for years. Clearly, also a wood business has been 6 years, and to meet new ones. Some of them we keep working online and from distance. We haven't had the opportunity to meet each other. It's a topic that I have chosen because for several reasons. First of all, I started working with the storage product as a drift contamination from period I spent in the Sosa in the vicinity of Raoul, so it was some kind of contamination. As he said, it is a system for really cheap system to work with, and it's almost a system for a lazy person to come. Sorry, I'm believing. But yeah, it's a work that we have been doing within a project that we established some partner, and where we have at least 2 students, one of PhD, another in as a master student leading with this project. As you may know, Brazil is one of the biggest producers of commodities, including soybeans, corn, wheat, rice, and during the process of storage of these commodities, commodity can suffer from huge problems, including problems caused by pest infestation. And the economic loss can be very high in terms of money. Okay. For this, generally, we have a number of pests that are frequently encountered in these commodities, and these sometimes processed food. It's very important to remember that most of them are actually from Coleoptera order, and some few ones from Lepidoptera order. And we have some mites also that can appear as problematic. And that's why probably, as I will say, a lot of works with bioinsecticide in this area are with Coleoptera insects. During a long time, the main control strategy is chemical-based one. And the problem is that there are really very few active ingredients that are used, mostly fumigant with phosphorine as a lead active ingredient used. And we know that the overuse of insecticide generally will result in the resistant selection of resistant population, which is a scenario that we have been experiencing in Brazil as producer and exporter. In this sense, we had established a project with some partner, including the University of Lavras, where I'm working, different lab from this university, but also some other institutional partner like Embrapa, who has long experience in terms of research, and especially Embrapa, Milho and Sorgo, which are really very focused on problems related to commodities like rice, corn, and so on. We included also some private companies that are willing to to embark in these experience. The idea is simplest. We try to revisit actually a lot of the existing alternatives. And that includes some inert dust, plant-based products, microorganisms, and we try to include some novelties, especially from the biotechnology side. Okay, and as Michele and Alberto said, when you are working with plant-based, you have to think of some kind of formulation. So the first line of research focused on zeolites as inert dust for the control of pest products. You may know that zeolites are naturally occurring minerals. They are crystalline with aminosilicates. And this is very interesting in the case of China also, because China is one of the biggest producers of zeolites in the world, which means that it's something that can be applied here. China has a lot of quite an impressive production of commodities also. More than that, zeolites are used already in agriculture. So they are used as soil amendment, they are used as ingredient to improve plant health. They are used for in husbandry as element, as feed complement also, and used in aquaculture to improve the system, the fishing system and fishing production. So there's it is a product that is well known and enough used. And besides being natural, we have also industrial production of zeolites. So this is why it was one of the products that we choose to test. And the idea was to take different zeolites from different origins, and also, when it is possible, with different size of particles, and test them as a system for range protection against different pests that can be found in during the storage process. We used diatomaceous earth as a positive control because it is already registered and used in many different countries. And of course, we had non-treated control. So first of all, we did some toxicity bioassays, really very short-term toxicity over 7 days, and it is a lab-scale bioassays where we treated different quantities of the grain, and we put different numbers of pests in contact with this treated grain. Then after that, it's really very short duration test just to see the acute toxical effect. Then after that, we try to see a much longer term bioassay, trying to understand what will be the effect in population growth. After that, we try to understand if there are any repellency effects. So these three types of bioassays were imagined and were designed to mirror the scenario that can happen during, before harvest, during harvest, and after harvest. So if there is possibility to kill the insects that are there, if some of the insects can escape the direct toxicity, if there is a possibility to stop the population growth, and also to avoid new invasion from different population. So the first result was really very clear, and we could see that from the different zeolites that we used, we had at least one which is Slovakian with 40 micron as a particle size that is matching the most used diatomaceous earth. And they could kill up to 75% of all the exposed insects. Then when we tried to look at the population growth at different concentration, we could see that beyond 500 ppm, the growth is inhibited. And nearing the 1000 ppm, which is the dose recommended for the diatomaceous earth, we are almost zeroing the population growth after 3 months, which is quite very interesting in the good. And then when we see the possibility of reinfection or reinfestation, we could highlight that from 500 ppm, there are few insects that can just survive the treated grains. So that was really very encouraging to go beyond this toxicological test. And then we tried to answer some curiosities on how this dust works. So we treated some specimens and we went through microscopy, and we could see that the contact of the dust just provoke or cause damages in ultrastructures the cuticle. Here to the right, you can see the non-treated insects with the syncytia that are really normal. Here you can see the rest of some dusts and the syncytia that are really very, very damaged. So one of the mechanisms that we think would be in action is just the mechanical erosion of these structures. Then we thought that since the Slovakian zeolites had this good effect on Cytophilus zeamais, that was used as surrogate for the effect on pests, we will try to test it in different other pests. So we used 4 other species: Triborium, Acanthocylidis, Cryptolithis, and Plogia, to see if they will be responding the same way. And of course, we could initially confirm the effects at 1000 ppm for 3 of the 4 new pests. We could see nice control, nice mortality, but unfortunately for Triborium castaneo, didn't work. And that was one of the surprises that we found. That's still under investigation. Probably we think that there are some differences in terms of the cuticle structure that could explain that, but we have not yet answered. Besides that, we went to a bit bigger scale. So we used that Slovakian zeolites. Actually, we used all the zeolites that we had, and we treated the higher and the bigger quantity of corn. Then we put it under normal and field condition, storage conditions for 4 months practically. Then we analyzed the damages from the different insects that could be present there. And surprisingly, we could see that practically all the dust that has particle size lower than 40 micrometers could keep the population and the damage really very low compared to the control. So that was the first step in terms of testing the alternative. We could see that the zeolites that we have been testing had a good effect, and clearly next step is just to try to see if we could replicate the same results at larger scale. And to solve one of the biggest problem with this kind of methods is the application of zeolites, especially right after the harvest. The second alternative that we tested were microbial, and we know that a lot of nematodes can have entomopathogenic effects. And this is why we used some Brazilian isolates that we have in our microbial bank to see if we could resolve at least the problem with Ribolium, who was less affected by the zeolites in this term. We tested standardized dose, which was 1,000 infective juvenile per milliliter, and we used two genus of nematodes, the Nerma and Heteroabitides. And we could test 11 isolates that were that are native to Brazil, that were gathered from different localities, and we could see that at least 4 of them had quite good impact, mortality to tree volume, but also to Cytophilus zea. And then we tried to see the effect of increasing doses, increasing concentration of infective juveniles in these 2 pests. And we could verify that Carpocapsi and Brazilians had quite a good dose-dependent effects, which is very promising. We are still testing different other isolates, so we continue the screening for efficiency of the isolate that we have. And as we know that nematodes, entomopathogenic nematodes, act actually as vector of bacteria, that is the killing agent, we are now at the stage of studying the microbiome of the 2 nematode isolates. We already identified the bacteria that are in the 2 species that were much more promising. We are at the stage of cultivating them outside of the nematode, which is really very, very hard to do and very challenging. But it's a work in progress that we hope we will be able to achieve in a few months. The last alternative, and I couldn't leave the wave and the fashion without talking about it. And here I will be confirming some results from Michele, from Alberto, but also from Raul. We have this, I think we have seen already this graph. We have seen a huge increase in the production papers related with plant-based products, and not only plant-based, but biopesticide. And a big proportion from these biopesticides are botanicals from different families. And within these botanicals, essential oils are very central as the ingredient, but also the isolated compounds. I am as critics in terms of using this botanical as Howard. We think that it's not because they are from nature that they are safe, and we should be very, very careful when we are using them. So this is why we start using actually isolated synthetic compounds that have been identified in plant essential oils, just to have the hydronized component and the component that we know the concentration and its purity. So we tested different phenyl propanoids and terpenes on the surrogate for we consider surrogate for pests and storage pests. and it is very easy test, very simple. It's a toxicity, contact toxicity test, and it is very short, 1 for 3 days. We've tested the anethol, anisaldehyde, cinnamaldehyde, eugenol as phenyl propanoid, and carbacril, carvone, and beta-citronol as terpenes. Except from citronol, that was not really very toxic for the test. All the others show really very similar toxicity and very good lethal action. After that, we just try to understand where is the effect, how it acts. So we tested, we went to assess the histological alteration that the exposure to this compound could provoke. And there was not a clear trend between the compounds that we used, but we could see that some of them can affect the muscle fiber thickness. The other one, the mid-gut thickness, which is structure for nutrient absorption. Others can affect the lipid reserves, which is the structure for energy storage. So depending the component, you can see different effects. There is no clear single target for this component. But some of them also could trigger interesting and positive reaction of the detoxifying enzymes and oxidative stress related enzymes, especially the anisaldehyde. We tried to make combination mixture, not to go back to the essential oils, but at least to try to test mixture that we decide what proportion is in the mixture. And here we tried all the mixture of the compost that we had under investigation, and it was always 1-to-1 ratio. And most of the mixture showed either synergetic or additive action, except for the mixture anisaldehyde and carvone, which was really very antagonist, which is surprising actually. Then, just to keep the phytotoxicity lower, but also to see the potential of extending the action, we nanoformulated this compound in folic caprolactone nanoparticle, and we could see that actually the nanoformulation extended the action of the terpenoids and the phenyl propanoids longer than the pure compound. Last but not least, we started few time ago to test the side effects in different organisms, non-target organisms. And here we used a parasitoid, which is not directly linked with the storage system, but that can be found in field, and it is used quite a lot. And we tested those compounds in different aspects of the biology of this parasitoid. And as you can see, the carbacril was a really negative effect in this parasitoid, and different compound could negatively impact not only the survival, but also the parasitism ability, emergence capacity, and even sometimes the total progeny. Interestingly, and here we are coming back to hormesis, one the one that was most toxic for the pest induced hormetic effects in survival of the parasitoids. So we went from 11 days as the median survival time in the control to 70 days, which is almost nothing. So yeah, natural compost can have both side effects. They can negatively impact non-target organisms, but also they can sometimes produce the hormetic effect that has been discussed before. Ongoing works include testing more compounds, increasing our knowledge about the selectivity of these natural compounds, because it's something that is lacking, especially in the manufacture. It's really very recently that many groups start taking this in account and improve the nanoformulation process of these naturally occurring compounds. And finally, we just started with the enzymes. We received 15 enzymes from different origins, and we just have been starting the process of screening them. And the LNI project is just to be done with Nico's lab, with our PhD student there to try to solve this problem. So this is what I had to tell you today, and I would like to thank all the partners and especially also all the team that has been working in the projects together. And also, thank you.
Discuss our conference. Last speaker is Dr. Conner from the University of Bavaria. Dr. Conner's research focuses on the ecology of insect vectors and their interactions with host plants and pathogens, which are critical for understanding and preventing plant disease outbreaks. Welcome.
So thank you. It's weird to talk at the microphone. Thanks for the invitation, and thanks to all the speakers that preceded me, because of you were very clear, while I will be not, because of my talk is about complexity. So let's start with this. When dealing with direct pests in crops, the most used approach is sort of the simple one. We use control tools to reduce the population density to an economically acceptable threshold. Okay, this is sort of a simple approach that can work for direct pests, but it has side effects. And secondly, if you're dealing with insect vector or plant pathogens in direct pests, this approach is never going to work. And this is sort of a demonstration. Actually, this is my case study for today. These are pictures from south of Italy, and this is a disease caused by a vector-borne plant pathogen, which is Xylella fastidiosa, is a bacterium. And actually, vector-borne plant pathogens are one of the greatest threats worldwide, against which we should find a control strategy. The problem with the simple approach against insect vector of plant pathogen is sort of not working. It's sort of triangle of the disease.
Your finger.
Triangle of the disease versus this. So our idea about usually the interaction between plant pest and environment is sort of a simple triangle with linear interaction. Okay, but if we just consider these, we overlook several aspects that are fundamental and that can offer us perspective for the control. Each one of these three elements, pathogen or pest, environment, and host, interact with each other and with other elements, creating a complex polygon. And actually, understanding this complex polygon is fundamental for us because we can really find solution for the control into the complexity. So responding to questions like how the pests interact with the environment, how the pests locate the plant, what are the traits that make a plant a suitable host for a vector, and how the different elements, biotic and abiotic, influence the pest. And in my case, the transmission of a plant pathogen is fundamental. So this is an hypothetical concept, and let's translate this into practice. And the case study for today, I already mentioned, is the bacterium Xylella fastidiosa. Xylella fastidiosa is a xylem-limited bacterium, which is a vector-borne, native to the Americas, and actually present in several countries in Europe and in Asia, and actually also in China. It was reported in Taiwan, and recently it was associated to the walnut seed scorch in the Xinjiang region. Hope the pronunciation is correct. And actually, the disease caused by this bacterium are very serious, including the olive quick decline syndrome in olive, the citrus variegated chlorosis, and the Pierce disease of grapevine, which is named after this man, Newton Pierce, which was the first in 1892 who described like disease associated to Xylella fastidiosa. The main vectors of Xylella are xylem subfeeding insects. Sharpshooters are the main vector in America and in Asian outbreaks, while spittlebugs are the main vectors all around Europe, and it seems also in North Africa. The main vector to olive of Xylella fastidiosa in Europe is this guy here, Philaenis fumarius, the meadow spittlebug. So how the meadow spittlebug transmit the bacterium to olive plant in olive orchard? Basically, you have juveniles that develop inside spittle on herbaceous on the ground cover, and then adults move to olive plants, and they transmit from olive to olive, a type of transmission which we call secondary transmission. Now, Purcell, you cannot see here, something went wrong, but it's fine. Purcell 1981 described this equation that you cannot read. Basically, the probability of the bacterium to be transmitted to a plant depend on 4 factors. The transmission efficiency, how much a bacteria is able to transmit the bacterium, the infectivity, which is actually the percentage of insects able to transmit the bacterium inside an environment, and then this combined parameter, NT, the number of insects on the plant per time they spend on the plant. We call this combined parameter vector activity. Actually, we characterize the transmission efficiency by Philaenis fumarius to olive and to grape. And the transmission efficiency ranges from 6% to a maximum of 20%, which could be a lot, but actually is not if you compare this value to transmission efficiency by American sharpshooter. They have a transmission efficiency of 90 to 95%. So in those cases, the transmission efficiency is very important. But for us, the most important parameter for the transmission of Xylella is this vector activity. So control is, how do we reduce vector activity? Well, in Europe, we tried a simple approach. We just try to decrease the population density of the vector through ground cover removal, which can have a lot of side effects against juveniles, and chemical control against adults. Results and surprise, the bacterium is still spreading. So the thing for us is we should find another solution because of the simple solution is not working. And this was a problem that actually drove us to start this journey for complexity. In my lab, we started to think that disentangling the complex interaction I was mentioning before is the only way to find alternative solution to control the vector, decrease the vector activity, and decrease the probability of transmission. So the first kind of interaction that we focused on is insect landscape. Basically, we wanted to know which kind of factors or combination of factors, landscape factor, climatic factor, agricultural practices, are the ones that are the most important for vector activity. Where the vector activity is the highest, the probability is the highest, and which factors shape this high transmission probability. So to respond to this question, we carried out a large-scale survey on more than 200 olive orchards that were sampled weekly over 3 years, collecting data on landscape, climatic variables, and also all the agricultural practices, administering questionnaires to farmers. So including data on pesticide use, which kind of pesticide, fertilizer, so on and so forth. And then we run basically 2 years of statistical modeling. And these are preliminary results from a random forest analysis. And this graph basically show the contribution of each variable that we consider on the vector activity. Surprise! The agronomic practices, ground cover removal, but also the pesticide use, contribute sort of marginally to the vector activity. The most important parameter for the vector activity are landscape composition, the percentage of semi-natural habitats, and altitude. And actually, altitude is not a direct factor for the vector. Our explanation for now is that altitude is sort of related with the water stress. The higher you go, the less the water stress, and you will see later why water stress is important. And during the survey, we collected also a lot of data on host plant preference for the vector, which was useful for us. We collected 3 years of data, for example, on herbaceous plants on which juvenile develop. So we ended up with a list of plants from the least suitable to the most suitable for the vector, and we are exploiting this data to try to develop a ground cover management based on the use of the least suitable plant rather than the complete removal of the ground cover. So this first part, insect landscape interaction, how we can translate this into practice. Basically, instead of having a generalized management plan, you can tailor the management plan, like applying certain practices only in area where you have a high risk because of the combination of variables is favorable for the vector and for the transmission of the pathogen. Second aspect of the interaction, insect-plant. The thing that I said before. Our idea is, what about the cues that drive the vector toward the plant? What are the cues that permit the vector to localize the plant? And second, once at the plant, what are the traits that make the plant acceptable for the vector? First, location of the host plant. As Mary is introduced, it's like host location is like communication in general is very complicated. For host location, usually insects use a combination of olfactory cues and visual cues. But the thing is that Philaenis pomarius is a polyphagous spittlebug, and 10 years of research more or less demonstrated that olfactory cues seem not very important for the middle spittlebug to locate the plant. But there was one overlooked aspect in host plant location, and this aspect is vision. Probably vision was at the base of the host plant location for spittlebug. And for this, we did a bunch of studies on anatomy, physiology, and ethological trials on the vector to characterize whether spittlebug were able to see plants and whether vision was important for host plant location. This nice picture is like a frame of a CT scan where you can see the aerodynamic shape of the eye. The eye of Philaenis is characterized by a low visual acuity, but actually Philaenis can see, and it's a trichromatic animal. Actually, it can distinguish color. It has 3 photoreceptors, sensitive in the UV, B, and green spectra. And actually, Philaenis is able also to perceive polarized light, which is very important cue to guide the insect toward the plant. And it's able to perceive polarized light in the UV and in the blue spectrum. And actually, through behavioral trial with in a Y-shaped maze, comparing dispersed and polarized light, we notice a significant attraction of Philaenis toward polarized light. Something that we are confirming right now through tests directly on plant, actually combining visual tests and olfactometer trials to exclude the role of olfactory cues. we are demonstrating that actually Philanus is not only able to distinguish different plants, but inside the same host plant species, these are olive varieties, is able to distinguish different varieties based on the degree of the polarization in the blue light spectrum. Sorry. What does it mean? That actually interfering with visual cue, most probably you impede the insect to locate the host plant, which is important. If you don't get the insect on the plant, you are not going to have pathogen transmission. But let's pass now to the other step. Once insect is on the plant, the question is, which kind of plant trait make a plant a suitable host for Philanus plumaris? There was very limited knowledge about this, so we decided to try to understand which kind of plant trait make a host is the most important for host acceptance by the bacteria. First thing that we did, we used the electrical penetration graph, the EPG, to characterize the probing and feeding behavior of Philanus. And then we tried to make correlation of the probing and feeding, so the acceptance of the host plant, with two plant traits, the anatomy of the plant and the anatomy of the xylem vessels, and the chemical profile of the xylem cell, because this insect is a xylem cell feeder. And we concluded that actually the most important trait for accepting the host plant is the right xylem sap chemical profile. If a plant has a right xylem sap chemical profile, it will be an acceptable host for Philanus plumaris. This is a data that we already published, but then we expanded on this data, like extending this analysis to multiple host species and host plant varieties, and we conducted an untargeted metabolomic analysis. We characterized the profile of the xylem sap, and then we did a bunch of stats, and we ended up finding the compounds inside the xylem sap that act as phagostimulant and phagodeterrent. I cannot reveal you this compound, but this will be submitted soon. So the conclusion of this is that actually, if you have a Philanus on a plant and you want to make the Philanus escape from the plant, you should manipulate the xylem sap chemical profile. No need of chemicals, because as you've seen from landscape data, the use of chemical does not really correlate with the vector activity. And we challenged this assumption with sort of a very simple trial. We took a plant extract, an Eucalyptus globulus extract, which has no lethal effect on Philanus. And we tested the effect on host plant acceptance of 4 administration routes. We sprayed the plant, we treated the bug, we did root drench, and we injected the compound directly into the xylem. This compound has no effect on Philanus in terms of lethal effect. And actually, we get no effect on the host acceptance, but for the direct injection into the xylem. If you inject this compound into the xylem, basically you reduce the feeding, you reduce the acceptance of the host plant, and you make the spittlebug escape from the treated plant, which has a direct reflection on the transmission of a plant pathogen because of, we did transmission tests with Xylella fastidiosa, and the transmission rate of the bacterium dropped compared to control plant. So if you manipulate the xylem sap with a compound which is non-toxic for Philanus, you get rejection of the host plant. But the use of exogenous compounds to manipulate the xylem sap is not the only way that you can manipulate the xylem sap chemical profile. And here we demonstrate this idea through water stress. Another important input, agricultural input, that is like important for the host plant acceptance is water. So actually, what we observe is that in some olive varieties, if you stress the plant, you actually increase the acceptability for Philanus plumaris. This is sort of the time that the individual stands feeding on the plant, which is longer on water stress plant compared to well-watered plant. When we investigated the reason why, we analyzed the xylem sap chemical profile, and what we realized, this is the severe stress. The chemical profile of the xylem sap is completely different between fully irrigated and stressed plant. And in case of stressed plant, they do not have those compounds that are phagodeterrent, the one that I showed you in the previous slide. Another important element that can modify this host-plant interaction that we should always take into account to predict the spread of plant pathogen are pesticides. So a pesticide can interfere with the host-plant interaction by the insect. What we did actually is, during the survey that I described here, we collected data about all the pesticides that are used on olive plants, including fungicides, which are usually overlooked, but are chemicals that actually can cause some sublethal effect on pests. And indeed, we tested like the transmission of Xylella fastidiosa after the insect were exposed to the chemicals topically and residually. And in both cases, for some of the compound, the molecular analysis here are still ongoing. For some compound, after the insect exposure to some compound, we got like higher transmission rate of the bacteria. So chemicals are important to consider in this equation because of they can really alter the pest-plant interaction. And obviously the pathogen transmission. And last but not the least, so today there have been a lot of talk on biological control agent, and mostly focused on consumptive effect, okay, direct efficacy of the biological control agent on the pest. But there are also what we call non-consumptive effect. Like actually, insect can perceive the presence of a threat, like a predator or parasite, using chemical cues and using the language of the insect, which are substrate-borne vibrations. And actually, the presence of the predator, consumptive and non-consumptive effect, can affect the behavior of the insect and can affect the interaction with the plant and the transmission with the pathogen. Since there are a lot of studies on chemical cues, we focus on the overlooked aspect, the substrate-borne vibration. And actually, what we observe is published here. What we observe is that substrate-borne vibration, like for example, the vibration produced by a spider walking on a plant, foraging for philaenes, explain most of the behavior of the insect, are not the only important component, but seem to be the most important component of the insect response to the threat posed by a predator. And we are actually like exploiting this concept to try to build a behavioral manipulation strategy based on substrate-borne vibration derived from natural enemies. These we recorded vibration produced by spiders, which are generally predators foraging for the prey or chewing on the prey. We used also vibration produced by ants to communicate with each other. And then we tested the effect, the disruptive effect on the plant-plant interaction of abiotic noises and also other vibration produced by heterospecifics, like other leafhoppers. And what we noticed to our surprise is that if you play back on the plant, vibrations associated only to natural antagonists, in this case, ants and spider, first of all, you change the within-host preference of philaenes. Basically, they tend to escape from the preferred portion of the plant and to colonize other portion of the plant. So they avoid what is usually the preferred portion of the plant. And secondly, they tend to escape from the plant. And the time that they spend probing and feeding, so ingesting the xylem sub, is very much reduced, especially in response to the ant vibration, which is fascinating because of, as we were talking about yesterday, ants are the first case of biological control adopted in China, more or less 2,000 years ago, less, but more or less. And at the end of this talk, I want to leave you with a take-home message, okay? And my take-home message for today is simplicity is better, but complexity is effective. We should not consider linear interaction. We should consider the system as a complicated polygon, and we should try to disentangle all of these interactions if we want to stand a chance, a sustainable chance, against pests, but especially against insect, bacterial, plant pathogens. And at the end of this talk, I hope was not too boring. So I would like to thank all the people that were behind these results. So my lab, people in my lab at the University of Bari, my collaborator at the Agronomic Institute of Bari, and all of the people and external collaboration that shared this fascinating, complex journey with me, the project that funded this research. And I would like to thank you for your attention, and I hope that this means CSCA and not other stuff.
From the landscape data that you show, you tell us that the altitude is an important factor. And then you show that the visual behavior of the vector is important. So do you think that the altitude is related to the UV radiation?
Yeah.
And this is important for the vector or for the disease?
Which is a super nice talk. So when I first had these results together with my ecologist colleague, he got excited. Oh, altitude explains everything. But I said it's not. Because of the higher you go, there is the UV, and the UV is important. The higher you go, you have less water stress, and you see that water stress can change this. It could be, I think it's more complicated than the single factor, but I think that altitude is just water stress, UV, all these other factors that like combine together, make the environment better for the bacteria.
Just out of curiosity, and regarding the vibration, it's easy to understand because spiders, that is extremely important for them. I was just wondering, and I remember in California, Rodrigo at the USDA there was doing research with spittlebug. Mating, it's also modulated by vibration. Just wondering if you had the chance to test this. Okay.
Yeah, these are just partial results. It's like we tested also the vibration, the intraspecific. The problem of mating interference with Philanocyte is super polyphagous, so you don't know where to interfere. Scaphoideus is on grape, it's fine. But Philanus is not. So I talked to the group, I talked also with Rodrigo Krugner about this. Intraspecific vibration, they have an effect, but it's not that huge. The thing is that it's like when you test the vibration of antagonists, they have this effect. The most fascinating part is that there is not the same effect caused by vibration that are intraspecific, or for example, Scaphoideus vibration, and the explanation to me is it's a flying feeder, it's not a threat. That does not compete. So there is something, and we are working on this, and also on aphids on the same topic.
Wanted to add something about the vibration. So if I understand well, you showed us some histograms with uptake on Xylem. But I think the reduction was not so high. When there was on vibration, I think it was 50%, something like that. Do you think we could use that, to develop a tool based on vibration?
Actually, the reduction is not to zero. There is still. It's we make sort of a count. If you have, I can talk more about this, but 100 spittlebug's on a control plant, 90% of them are theoretically able to transmit. If you have that vibration out of 100 spittlebug's, more or less 30% of them are able to transmit. At the same time, I don't think that vibration alone can be a control tool. But there is an interesting part, which is vibration causes stress in pests. So actually, vibration can make the pest more exposed to other stressors, which we have seen in aphids. So this combination is probably what we will try to apply in the framework of a European project that just started.
So it will be used in combination?
I think so. Okay. I don't believe that that single tool will make a lot.
Thank you.
to the next lecture is Dr. Head from the Tiger River organizations. Dr. Head's research integrates agroecotophagy, landscape ecology, and biodiversity conservation, particularly focused on the role of flower strips in supporting beneficial insects. Welcome, you have 30 minutes.
Thank you very much, Dr. Dab, for the very kind introduction. Before all, I'd like to thank very much the organizers of the conference, Professor Wang Su and Dr. Nicolas Deneuve, for inviting me this year again for this symposium. It's always a great pleasure for me to be back to China. And I'd like to talk to you today about flower strips and how they can be one tool in the IPM framework and within IPM packages. And I'd like to focus on from using flower strip from supporting natural enemies towards biological control. So indeed, I work for an organization in the French-speaking part of Belgium called Natagriwal. So who we are? We are an organization that advise and accompany farmers to improve the environmental quality and to improve the environmental conservation in agricultural landscape. We are not for profit and mostly funded by the local government. And what we basically try to do is to develop such kind of diversified landscapes. Why? Because we know that when we have more semi-natural habitats and at the same time, or as a correlation, a reduced size of agricultural fields, then we have more biodiversity. But what is also interesting is that among the biodiversity that we can preserve in such diversified landscapes, we have beneficial organisms that can contribute to the regulation of insect pests. We have a diversity of tools to propose to farmers to diversify landscapes, and among them are flower strips that are subsidized by the European Union and also by the member states, so the regional government. And we have in our region in Belgium different types of flower strips. We have, we have perennial flower strips. So they are actually made of perennial flowering plant species, typically those that we find in permanent pastures. They are implemented for a minimum of 5 years. I mean, this is the contract with the farmer, but actually they are sown once, and if they are properly managed, they can be kept for years and years. And then we have annual or bisannual flower strips that can be sown in either in spring or either in autumn, and the composition of these flower strips are different, but they are here made mostly of annual or bisannual plant species. You can see that these flower strips are subsidized. So the farmers, when they sow these flower strips on their field, they receive money from the government, and that can be quite a lot of money. For example, this perennial flower strips, it's 1,800 euro per hectare of flower strip per year. So I calculated it's about 1,000 renminbi per mu per year. And for these annual flower strips, it's about half of this amount. And of course, it's a powerful tool to get farmers on board towards conservation biological control. However, what we try to do is to tailor this measure to favor natural enemies of pests towards
pest control in the adjacent field. And yes, because it's important to understand that these flower strips were at the origin not designed for conservation biological control. They were designed to support biodiversity in general. And of course, there are some natural enemies. But what we try to do is to tailor the mixtures to have more natural enemies benefiting from these mixtures. And so to do so, to select the proper plant species. However, it's important to understand what drive natural enemies in flower mixtures, what attract the natural enemies in the mixtures, and what contribute to their conservation in the flower mixtures. So in an experiment I conducted, this was part of my PhD project about 10 years ago, we had different flower mixtures, and I was interested to understand what are the plant traits that actually explain the population of natural enemies that we find in these different flower mixtures. So what we did is to monitor the flowering biodiversity we had in the different plots, and at the same time we tracked the insects, and we described the different plant species we could observe in the different mixtures through different plant traits that I listed here. So the color, the ultraviolet reflectance, the ultraviolet pattern of the flower, the morphology of the flower, the height, the blooming time, and the blooming duration. And then through redundancy analysis, we crossed the community-weighted mean of the plant community, but described through their traits with the abundance of different natural enemies that we collected in these mixtures. And what we could see is that different flower traits drive different family of natural enemies in these flower mixtures. So we could see that holoflies, and this was mainly Epicirphus palteatus, was more important in mixtures where we had a higher value of flower with open nectar. So we've seen with Felix Wakers this morning that, yes, indeed, the availability of nectar that depend on the corolla morphology is a very strong driver for some natural enemies, especially holoflies. And for ladybirds, it was more viable, but we could, for example, see that the Asian ladybird, Harmonia axiridis, was more abundant in plots where we had ultraviolet pattern. So it means that the ultraviolet reflectance was different at the periphery of the flower compared to the center of the flower. We also looked at this effect of flower traits on the parasitoids of the pollen beetle, so the meligete species that are a main pest of oilseed rape crops. And we could see that the reflectance of the ultraviolet reflectance, but also the color, and especially the yellow color, favored the abundance of these parasitoids. And it's quite interesting regarding yellow flowers, because when we know that these parasitoids will actually target meligete larvae that are on the yellow flowers of the oilseed rape, then we can understand that actually the yellow flowers in the flower mixture is also an important driver for their abundance in the flower mixture. So we've seen that there are different flower traits that contribute to favor the abundance of a diversity of natural enemies in the flower strips. Another parameter that is important to consider for not the attraction but the preservation of the natural enemies in the flower mixtures are the presence of prey, or what we can call alternative prey, because they are not the prey that are the pest of the crops, but prey that are in the flower mixtures. For example, here on this picture, we can see a common veg that was part of this mixture, where we had aphids here and 3 ladybirds on this plant, just probably looking for having these aphids as a lunch. So here, this was an annual autumn sun flower mixture. So we had 5 species: mustard, forage radish, buckwheat, common veg, and sunflower. And so we monitored the flowering coverage here on 9 of these flower strips. But we also looked at 5 plants of each of the sown plant species, whether they were having these aphids that could be used as prey for the natural enemies. And of course, we also collected the natural enemies in the flower mixtures. And what we could see is that more than the variation of the flowering coverage, the presence of these alternative prey was explaining the total abundance of the natural enemies in the flower mixtures. So the more we had the presence of alternative prey on the sown plants, the most we had the numbers of natural enemies in the flower strips. And also diversity. So we had a higher Shannon diversity where we had more plants infested with alternative prey in the flower strips. And for the Simpson diversity, it had a marginal significant interaction with the flowering coverage, and the effect of the presence of the alternative prey was especially strong when we had also a high flowering coverage. And finally, the presence of the alternative prey reduced the evenness of the community of natural enemies. And this is quite interesting because we know from other studies that biological control can be driven by a reduced evenness, because it can favor some key natural enemies that will be especially effective to control the pests. But so then what happened in the crops adjacent to this flower strip? So here we studied these flower strips adjacent to winter cereals, especially winter wheat, and we counted aphids at 5 meters and at 50 meters from these flower strips, and we could observe a strong effect of the flower strip at reducing the abundance of the aphids in the cereals at 5 meters compared to 50 meters. But also interestingly, at 5 meters of the flower strip, we could stay below the economic threshold for aphids on cereals, while at 50 meters of the flower strip, we were above the economic threshold, that is 1 plant over 2, so half of the plants infested by aphids on cereals. We know actually that flower strips, but also hedgerows, have a strong distance decay effect on biological control. This is a meta-analysis published a few years ago, showing this negative exponential reduction of biological control adjacent to flower strips or hedgerows, showing that at about 25 meters from the flower strip, we actually don't have any positive effect of the flower strip on biological control. So having just a flower strip at the margin of a 10-hectare crop field would have a marginal effect, or at least an effect at the margin of this plot. So one way to solve this issue is actually to implement in-field flower strips, or, I mean, the idea is the same, that reducing actually the size of the field, the crop fields. So here it's not an experimental field, but it's a field of a farmer. So it's very interesting what this farmer did. Here's about a 10-hectare field, and some years ago he decided already to divide this field into two parts by sowing these quite large perennial flower strips. So these flower strips that were at the very beginning of the presentation here, right? This 5-year flower strip that is there permanently. And then last year, so for the cropping season 24-25, he implemented this annual flower strip that we see here, and also around the field. And for him, it was also calculated because he grown sugar beets in this field. And in the farmers, they have contract with the sugar company, and they have contract for a certain area of sown sugar beets. And this year, the sugar company said, we will not pay or not pay much any extra hectare and any hectare that are beyond the contract. And this field was more than the contract that this farmer had with the sugar company. So he thought, instead of growing sugar beets that the sugar company will almost not pay to me, I will plant this flower strip and also ask for the subsidies. But interestingly, he designed these flowers, so the field with the flower strip, with a distance of 27 meters between each flower strip. This is the size of the sprayer. Of course, the objective was to not spray insecticide, but the farmer will continue or had continued to spray herbicides, for example. So he adapted his design to his machinery. So 27 meters to be able to spray the herbicide especially, and also space to turn around here the flower strips so that the logistic of the mechanical work is feasible. This was another field where here also it was set 27 meters with a flower strip every 27 meters. And so here in these fields, so with sugar beet, we monitor the abundance of the pests and the natural enemies. So it's important to know that in Belgium and in Europe, we have an issue on sugar beet. That is, since neonicotinoids are not allowed anymore to be used in field crops, we had the re-emergence of the transmission of the beet yellow virus by these green peach aphids, or Mysis persicae. But the sugar beet is also attacked by the black bean aphid, Apis fabae. And we try to find solutions to solve the infections of these aphids on sugar beet. And so by using the flower strip, we monitored whether we could significantly reduce the infestation of these two aphid species on the sugar beet. And we could observe that where in the field where we had these flower strips every 27 meters, we could significantly reduce the abundance of the black bean aphid, at least until the end of May. At the beginning of June, the population increased. However, from here, we consider that the sugar beet are not that sensitive enough, are not that sensitive anymore to the aphids. So with the flower strip compared to control plots where we had no flower strip, or at least where we could go 50 or 100 meters from the margin, we had a significant reduction of the black bean aphid. However, for the green peach aphid, Mrs. persicale, we could not observe such a strong effect at reducing the population. You should notice that we had about 10 times less green peach aphids compared to the black bean aphid. And this is a discussion we had a little bit at the lunch. Is that when we look at the natural enemies, we had actually more natural enemies. So they are mostly there were ladybirds larvae, also hoverfly larvae, and some others like Ontochoridae or Staphylinnidae, but mostly ladybird larvae. We had also more of these predators at 50 meters, so in the control plots, than in the plots where we had the flower strip. And when we look at a potential correlation between the abundance of the natural enemies and the abundance of the aphids, we could see that the abundance of the natural enemies was strongly driven by the abundance of the black bean aphid. However, it was not the case for the green peach aphid. So indeed, if we have 10 times more aphids, black bean aphids, than green peach aphids, then the natural enemies were driven by the black bean aphids. And what may happen is that actually, with such a high amount of black bean aphids, the natural enemies will focus on them, and the tiny or less abundant green peach aphid may circulate and, I'd say, go hidden in this system and continue to be a potential problem without being significantly controlled by the natural enemies. However, the problem with the green peach aphid is the potential transmission of the virus, and we had amazing talks about whether the vector and why it does explain that the vector transmit or not the virus. Here are some aerial pictures of the fields we followed. And here is a field where there was one insecticide treatment when we reached the economic threshold of the aphids, but I could obtain that in the center of the field we had an insecticide-free zone. And actually, we don't see really any difference between the two, right? So when the virus is transmitted, we have yellow plants, and here we don't really see. Here we had no insecticide. The farmer decided, I will not spray this year. I don't want to spray. So we have some patches of the virus being transmitted to the sugar beet. Here we had one spray, also at the economic threshold. We also have some patches of the virus transmitted. And here we had no spray and we had no virus. So I was very happy to be able to discuss and to listen more on the factors affecting or explaining the potential transmission, because in this case here, the strategy is spray against the vector. However, spraying against the vector may not prevent the transmission of the virus, but at the same time, with flower strips or other landscape-based management, it might also be challenging to control the abundance of the vector. So probably we should go more in understanding the factors explaining the transmission of the virus in this case. So it's a lot of food for thought for us, for our case in Belgium. Another potential strategy is, and we had examples already of combining And here also, for plant diversification, we can combine different techniques. And one of it would be to combine the flower strips with crop diversification. And one way to diversify cropping is intercropping, so that is the cultivation of at least 2 plant species simultaneously. So in this experiment, we had intercropping over 2 years. The first year we had fava bean intercrop with winter wheat, so we had a half density of wheat added to a full density of fava bean. And the second year, we had red seed poppy that was intercropped in the same way, but with barley. And we had plots with flower strips or without flower strips. And we had also each time the pure crop as a comparison. And we were looking whether we had any combined effect of intercropping and flower strips to strengthen the regulation of aphids on these two crop years. And what we observed is that first, intercropping was the stronger method to reduce the infestation of aphids on both fava bean in the first year and breadseed poppy on the second year. So it's interesting to see, and you might know that intercropping is a strong method to control aphids through 2 mechanisms. First is the risk of the inappropriate landing by the aphid, because if the aphid of the black bean, of the fava bean, so black bean aphids, for example, land on the cereal, and it will land on the non-host plant, and we'll have to continue to search for the host plant. But while searching for the host plant, he would face the presence of the non-host plant again. But also, while spreading, he would face the non-host plant, so the non-host plant would also play the role as a physical barrier controlling the spread. So here in our case, intercropping was consistently controlling or reducing the infestation of the aphids on both the fava bean and the breadseed poppy. And the second year, we could also observe significant interactive effects between intercropping and the presence of the flower strips, but also the distance. So we could see that at 10 meters, but not at 20 meters, we had even stronger reduction of the infestation of aphids on breadseed poppy. Right. Actually, I run at the end of and just the take home message. The take-home messages is that food provisioning for the natural enemies through nectar availability, but also through alternative prey. And we have the example of the cover crop in the orchard in Spain. The alternative prey are also very important, are also a very important driver of the presence and abundance of natural enemies in the flower strips, but also some attracting traits that are visual traits affecting the behavior of natural enemies would contribute to attract and preserve the natural enemies in the flower strips. However, we need new field arrangements because we know that there is a strong distance decay effect on biological control. So just having a flower step at the margin, we will not solve most of the problem. And one way also for new field arrangements is also to combine different approaches of diversification. For instance, by also having mixed cropping in addition to diversified margin in agroecosystems. This is what we try to do with farmers. We organize field days, field visits, and farmer-to-farmer meetings where the farmer, for example, this farmer who had this very interesting design showed to the other farmers what he did and how he did it, because farmers trust other farmers more than scientists sometimes. And we also organize training where we try to explain, however, as scientists, some mechanisms. I think it's also important to explain to farmers what is behind the effects. I'd like to thank the collaborators for this research in Jean-Bleu Agrobiotech with the laboratory of entomology, where Dr. Qingxuan Xu was also last year and helped me to collect some of the data I showed you, and also where I did one of my postdoc research at the University of Bonn with Thomas Doring. Thank you very much for your attention, and I'm happy to discuss with you. Maybe we have 1 or 2 more minutes.
You know, I got excited when you said aphids. So the first thing is just some thoughts about, have you ever used plant traps?
Yes, we use plant traps for, so here not for monitoring the aphids, because the aphids we count them on plants, but we use the plant trap in the flower stage.
What about you use the plant traps for monitoring dispersing aphids to see what are the species that are related to the BTL?
So what is your idea behind this? That there could be
it could be means or could be just a transient species because of your pattern is very weird.
So, but in this case, we should have, because the yellow virus, we see this 3-4 months after aphids were in the field. So we should cover the field with plant traps and then see where the virus, like this yellow rounds, and try to see in which plant trap this yellow round is, and then look at that.
At least characterize which species is virus. At the beginning. At the beginning, I would not care about the virus. I would care about transient species. What are the transient species? Okay. And the second thing is, what's your hypothesis? Is that you showed us different situation where you have virus and you have no virus. There could be that when doing chemical treatments and when there are natural enemies, there is disturbance so that the aphid move and then you're going to get your transmission. Is a reasonable hypothesis something you thought about?
I don't know, but if you think that is a reasonable hypothesis, that actually the treatment will favor dispersal, it's very interesting.
Yeah, it's something that is described.
Okay.
The treatments favor dispersal. As natural enemies, they favor dispersal.
Yeah, natural enemies I know, but treatment, then...
Yeah, they are disturbed, so they even probe more, and you have a higher frequency of transmission of plant viruses. Could that happen?
Yeah, that would be interesting, because last week we had a meeting with different actors of and this was on pesticide in general, but also the Technical Institute on sugar beet. And the message was, there is no choice, we should spray. So, but when we see these pictures, we say spraying does not explain the patterns of the virus transmission. So any arguments that can maybe bring more complexity in this kind of very simple message saying we should spray, we have no choice. And I'm very interested.
Try to bring this message. We'll back you up.
Yeah, thank you.
Speaker is my colleague, Dr. Jining from BASS. Her works explore the complex interaction between heavy metal stress plants, polyvirus pests, and their natural enemies. We have 20 minutes.
Okay, thank you. Hello, everyone. I think I will face my laptop. I'm Ningdi from BAAFS. Today, I'd like to share some of our work about cadmium-mediated multi-trophic interactions between plants, pests, and natural enemies. So firstly, I will start with a brief introduction of myself. I work on natural enemy breeding technology, and I also do some work on the interaction between insects and plants. I started working on heavy metal regimes from 2014 when I was visiting UC Riverside. Today I will share two case studies from our lab. So starting with a brief introduction. So agriculture ecosystem is actually, I think this afternoon some professor introduced this system is a very, it's a system with multi-factors that are very important to this system. For the crops we grow is centered with crops and including the herbivores, natural enemies, and the surrounding environment. Previous research has shown that many biotic and abiotic factors can break the balance of this system, and among which heavy metal pollution is considered a very important factor as a regime that affects the multi-trophic interaction. For the heavy metals, cadmium has become one of the top polluting elements around the world. As we all know that cadmium threatens the environment, and it also has toxicity such as alters genetic material stability and reduces individual fitness for the insects. So, as a prevalent and significant abiotic stressor, cadmium exerts bottom-up cascading effects, influences trophic interactions between plants, herbivores, and natural enemies, thereby may impact the efficacy of pest biological control. This is the wrong version. That's okay. This is the wrong version of my PPT. So there are two hypotheses that leading this cadmium research work. The first one is the induced distance hypothesis, and the second one is the passive selection hypothesis. So here we wanted to test what principles govern the cascading effects of cadmium from pests to their natural enemies, and how might this impact the predatory or parasitism efficacy of natural enemies. So we worked on two systems. The first one is the predatory stink bug, and the second one is tetragram wasps. So firstly, I'd like to introduce the predatory stink bug, Oryx sautere. Oryx sautere is the most widely used predator in China for controlling thrips in the field, both in the open field and in the greenhouse. So firstly, we worked on this system, the copy Western flower thrips and Oryx sautere trophic system. We assessed the fitness parameters across all three trophic levels and preliminarily explored the underlying behavioral mechanisms. We found that the fitness of Western flower thrips decreased on cadmium-treated poppies, and also thrips mortality increased with the rising of cadmium concentrations. Then the predation rate of Oryx sautere showed no significant difference in the short term, but most interestingly, the Oryx consistently preferred pre-infested copy plants over the cadmium stress. So they choose preys indiscriminately. They can't detect if the hosts are stressed by cadmium or not. So while the heavy metal cadmium stress on copies reduces thrips fitness without affecting natural enemy foraging behavior in the short term, it elevates the risk of passive cadmium accumulation in the natural enemies, potentially dismissing their efficacy in pest control application. Then we worked with the tomato system because the tomatoes, especially in the last 5 years, it faces severe damage by the thrips in Beijing. So we wanted to detect the cadmium content across the three trophic levels and measure the fitness indices of each trophic level, and then do some investigation into the behavioral mechanisms involved. So firstly, we found that the cadmium stress influenced the accumulation of cadmium in various tomato tissues. The roots, stems, and leaves, all of the organisms accumulated significantly high amount of cadmium, and the population structure was then changed of the Western flower thrips by the cadmium treatment. And most importantly, the population structure is primarily mediated through the suppression of female adult reproduction. And we found the same trend in the whiteflies. However, we also found that the pest fitness, the decrease of the pest fitness is associated with accumulation of heavy metals. All treatments of the Western flower thrips accumulated significantly high amount of the cadmium compared to the control. And we calculated the transfer coefficient and the bioconcentration coefficient. We found that both of them from the plant to this pest were above 1. So the cadmium accumulation plays a decisive role in reducing the pest fitness. Is this the only contributing factor? The answer is no. We also found that the decreased pest fitness may also be mediated by the accumulation of secondary compounds. So we found that 5 of the compounds tested, they showed varying degrees of suppression of the pest survival, and then we conclude that cadmium exerts a dual toxicity effect. First one is cadmium-mediated plant-induced resistance by generating all of the secondary metabolites. And the second one is cadmium bioaccumulation and its direct effects. For the foreign behavior of oris, we found that their choice behavior, the behaviors are determined mainly by the prey ability. The same with the prior work we found on the copy, as opposite to the presence of cadmium stress. So they choose the prey discriminately, even if they are stressed with cadmium. Also, the predation amount of oris solitary was initially showed no difference, but it significantly decreased after 24 hours, indicating feeding suppression. Despite showing no correlation with, where is it? Oh, okay. So the away position of oris solitary significantly decreased after feeding on prey from cadmium-stressed plants. This raises the question: is the decision induced by the internal accumulation of cadmium in the natural enemy? The answer was yes. The answer is yes. So the natural enemy accumulated a high amount of cadmium. So the reduction in natural enemy fitness is also associated with accumulation of heavy metal. We then calculated the bioconcentration coefficients and the transfer coefficient. We found that although the transfer coefficient from tomato to pest to this natural enemy is below 1, but the bioconcentration coefficients were all above 1. This indicates further toxicity to the natural enemy. Then I'd like to talk about the other case study, cadmium-mediated pest-parasitoid interactions. Cadmium pollution in paddy fields affects the growth and the development of rice pests. Previous studies found that the roots, stems, leaf, and grain all can accumulate the cadmium to a certain amount, and consequently, the development feeding on the rice can be adversely affected. The Trichogramma wasps are currently the most extensively studied and widely applied egg parasitoid insects for controlling the rice stem borer. With a big family and with a big range of hosts, it has been used in China widely. For example, the annual application of Trichogramma dendrolimi for controlling the Asian corn borer exceeds 4 million hectares. Consequently, the Trichogramma wasps face a high probability of cadmium exposure. So we wanted to test the impact of cadmium transfer through hosts on the parasitic fitness of Trichogramma. Here we used 2 species of Trichogramma, the dendrolimi and the japonica. The first one is has a wide host range, but the second one has a narrower host range. We found that cadmium stress significantly inhibits photosynthesis in rice and adversely affects various growth parameters and causes leaf yellowing and plant death in the later grown stage of rice, and then it inhibited dry matter accumulation in rice, and resulting in notably reduced dry weights of roots, stems, and leaves. Also we found that the stems accumulated considerable cadmium, and the accumulation level increased progressively with the duration of treatment. For the pest rice stem borer, when they feed on cadmium-stressed rice stems, they show a significantly reduced larval body weight, while the adult longevity also decreased, but we didn't find the statistic significant. But interestingly, the second instar larvae of rice stem borer exhibited a significant preference for cadmium-stressed rice stems. Then we tested the parasitism of the 2 Trichogramma species. We found that cadmium did not affect the parasite fitness of both species. When not exposed to cadmium stress, Trichogramma wasps showed no significant parasitism preference. However, cadmium stress induced a significant preference for the parasitism, for a significant preference for parasitising cadmium-stressed hosts. Sorry, I think this is a wrong word. It needs like 30 minutes to finish, but I will be fast. Okay, we found a very interesting thing that the host cadmium stress enhances the competitive ability of this host japonica with a narrower host range. Okay, then we want to see if the cadmium stress affect the host habitat location. We found only Japonica showed preference for cadmium stressed rice plants. We then identified 9 compounds from cadmium stressed rice that showed significant difference, and we tested the preference. We found that this compound with a long name, the name kills me. It shows it attracts only the Japonica. Then we stress the tetragramma wasps with cadmium. We found their behavior was altered, and then still this interesting compound exhibited a concentration-dependent effect on tetragramma japonica. We also did some like morphography stuff and locomotion of tetragramma wasps. And we also found that the cadmium accumulation only significantly increased in tetragramma japonica. So I think I will not read a brief summary. And I think in the future we need to do some more field work and establish a heavy metal pollution gradient observation network with the monitoring population dynamics of pests and natural enemies at a regional scale. Okay, and then our work is funded by this funding, and we thank our collaborators, Professor John Trumbull and Professor Nico, and this is our working team. Okay, thank you for your attention.
Speaker is also my colleague from Dr. Wang Haiping. Her research was focused on the molecular plants pathology, particularly the regulatory mechanisms of function, pathogenicity. About the mechanism of LT958 cross-candom regulated pathogenicity of Lasodiploides cerebromium. My name is Cai Binfang. I'm an assistant researcher from the Beijing Academy of Agriculture and Forest Science. My main research is focused on the grape chemical disease. So as you can see from the slide, the grape chemical disease actually belongs to one of the grape trunk disease. It's caused by the plant fungi and leading to significantly yield loss in the grape industry and cause big damage in the Chinese grape industry. And the symptoms of the grapevine chemical disease includes when the grape is during the graping period, the ridges will dry out and the fruits will draw, and the branches will ulcerate. In several cases, the whole plants will wither and die. So the main pathogen of this grape chemical disease is called Lasodiploides cerebromium. It's very difficult to control this grape chemical disease. There are several reasons. The first reason is because the grape chemical disease, the Lasodiploides cerebromium, is the major affect vascular boundaries of the grape. And second reason is Lasodiploides is an endophyte and polykaryotic fungi in grape branches with a long incubation period. And the family reasons is because this fungi, most of the time, is to stay in the plants. In some certain cases, like the temperature increase, it can transform from an endophyte state to a pathogenic state. And this mechanism, this pathogenic pathogens we didn't, at least remains is unclear. And when we study, because today there are many insect, very professional insects, but this fungi is transmitted physically. It's not transmitted by insects. So I will only focus on the study of this pathogenicity of these fungi. So in my studies that my mainly focus on the microRNA, like RNA. As anyone knows that microRNAs are endogenous small RNAs of 20 to 24 nucleotides. And the microRNAs, the main regulations for either in fungi and insects and in humans, that is regulated target mRNA's expression by inhibition, the translation or transcription silencing of the genes. And in fungi, there are part of the small RNAs is very similar to plant and animals and insects for the mRNA like RNAs. So we write it like a mRNA. And many people in study of the mRNAs, because they can create the biocontrol agents targets of the mRNAs, they have different ways to control, to use these mRNAs. One is by spreading induced gene silencing, is by targeting these RNAs. These RNAs is emulated by the nanomaterials and then spread on the crops and to against the plant pathogens. And the second way is we do the transgenic plants, which can stably expression sRNAs. So this is another way to use to control these plant pathogens. And now it's very new ways is that called the microbiome-induced gene silencing, is that we use some bacteria that continue expression those small RNAs and together with some other microbiomes to form microbiomes to spread in the soil of the plants to control these plant disease. So this is we are focused on to do this research. And now in the world, there are many commercial RNA-based biopesticides, but most of the biopesticides is actually insects. As you can see, is very commercial ones. There is no commercial RNA-based pesticides to control plant pathogen. So this is we are focused on to study in this part. And in my research, we would like to first to identify mRNAs that regulated the pathogenicity of this Lassodiplodia, and exploring the pathogen mechanisms, and then provide targets for RNA fungicides. And at last, we would like to prevention treatment of this great white canker disease. In my research, in study of these fungi, we first do the small RNA sequence of these plant fungi. We do 4 samples. One is just mycelium cultured at 25 degrees, and also that mycelium cultured at 37 degrees. And a second is under the infection conditions, is that mycelium cultured with grape bark powders at 25 degrees and the mycelium cultured with grape bark at 37 degrees. We want to see some genes, some significantly induced genes during these 4 samples under infection condition. And interestingly, we found that MARA55 is significantly upregulated after infection. So we continue to study whether this microRNA in these fungi can regulate the valence of this Lassodiplodia. And interestingly, when we overexpression these small RNAs, we can see the infection area are much larger than the wild type. And also in 37 degree that the infection area also larger than the control plants. So means that this microRNA 58 is positively regulated the balance of this Lactobacillosis. And very interestingly, when we try to find the structure of the microRNA 58, we can see that this microRNA 58 is formed in stem loop structures, and it's very similar with the grape. So this is from the fungi, this is from the grape. The grape microRNA 485 has a high similarity with the fungi microRNA 58. So, and very interesting, for the grape, microRNA 482 is target 25 NR genes that can specifically inhibition these plant immunity genes. As we know that microRNA 482 is target these 28 genes and are core component of the plant immunity systems. So based on these backgrounds, we think whether the fungi microRNA 58 can cross condom, so from the fungi to regulated grapes and our genes, resistant genes in grape to influence the pathogen process. Based on this, we do some experiments. We just overexpression microRNA 58 in the grape leaves. We want to see whether microRNA 58 from the fungi will affect the plant immunity. And when we overexpression the fungi genes in plants, we can see it's inhibition the whole growth production, and also can inhibition the callus stabilization, which is important in plant immunity. So means microRNA 58 can suppress the host immunity response. And secondly, because it's a half size, we need to experimentally to confirm that whether this fungi microRNA 58 can, from the fungi cross condom to the grapes to affect its plant immunity. We do one of the experiment is we use one of the plant pathogen infection samples, because the fungi and the plant cell walls has different components of the cell walls. So we add cellulose, pectinase, and hemicellulose to specifically digest the plant cell walls, but not digest the fungi cell walls. And after we got this plant protoplast, we want to detect whether in those plant protoplast there existed some microRNA 58 inside of the plant protoplast, but there is no existed of the fungi materials. So we do this part of the experiment. And finally, we found some interesting things. In these samples, in the plant protoplast, we found the fungi microRNA 58 genes, but we do not detected any of the fungi materials. So means this microRNA 58 can successfully transform from the fungi to the plant to the grape. And then we want to know whether this fungi microRNA 58 to regulate those grape genes. So with some of the website, we try to find some potential target genes of these microRNA grapes. We found 7 genes of these grape genes. And by experimentally, we use this agrobacterial fluorescence experiment. So we co-express of the microRNA 58 with those target genes with GFP tag. And under this 7 genes, we selection some interesting genes like these genes. We can see when it co-expressed with 58, the GFP intensity will be lower than it expressed alone. So this means that microRNA 58 is inhibition the expression of these genes, and it is the target. So finally, we want to know whether those target genes in the grapes, whether it were affected the plant pathogen infections. We do some transgenic grape leaves with these genes. Now is doing the genetic transformations, and later we will continue to do the infection experiment. So these are all my research stories. I would like to thank you, my team, Integrated Disease Control Laboratories in Plant Protection Institute, and another 2 PhD candidates, Wang Yuan and Italy, and also thanks the National Grape Industry, and the NFFC support. Thank you for listening. Okay, the last year speaker is Dr. Man Shi. She is a postdoctor in the Applied Entomology Lab. Research was studies of functional plants on the insect management.
Hello everyone. I'm Ma Chi, a postdoctor member from the onSustainable lab. And today I'm very honored to share in the report our team research. And this PowerPoint I show is the work from Associate Professor Li Shu and her students. And the topic is about the application of functional plant management technology in biological control of pests in protected facilities. And my presentation is divided into the following 4 parts. With the acceleration of urbanization and agriculture modernization, people's concern for food safety has increased. And as a result, the greenhouse product market is showing a great potential. And the global certified organic farmland has reached 70 million hectares. And according to the prediction of, and it's showing a continuous growth trend, and according to the prediction of relevant organizations, Asia's market is expected to increase significantly in the future. And the protected agriculture is facing severe pest problems. The most severe pest species were whiteflies, leaf miner, aphids, and thrips. And according to the GBF data, we can find that the occurrence of these pests are global. And these kinds of pests often happen consecutively and spread rapidly, cause significant damage to the vegetable or crop. And traditional chemical control methods present numerous issues like pest resistance, ecological imbalance, and food safety concerns, even the risk to human health. And traditional mass release technology for natural enemy suffered from high application cost and significant associated risks, and even a lack of integrated technologies. And hence, there is an urgent need. There is an urgent need for new biological control approaches in the By contrast, cultivating functional plants in the facilities enables the establishment of relatively balanced agricultural ecosystem through the interaction between pest, natural enemy, and the functional plants. And this approach can also develop a low-cost and highly efficient and sustainable pest management technology. And we must know what is the functional plants. Functional plants can provide natural enemy with food, shelters, and overwintering or breeding sites. And these plants can also provide suitable microenvironments for natural enemy growth. And take the case of the Cnidium in the current wheat rotation ecosystems an example. And we can find that it can provide little beetles with functional service like conservation, migration, and transitional. And then this functional service were throughout the year. And there are many names or types of the functional plants. Many include the following categories like honey plants, trap plants, repellents, combination, habitats, and backer plants. As the name implies, plants with different functions may possess distinct mechanisms that enable them to have effective influence on their natural enemies and thus control the occurrence of pests. And the core mechanism involved the bottom-up effect, top-down technology, post-pull systems, and the attract and reward methods. And these systems for technologies were based on theories such as the natural enemy hypothesis, resource concentration hypothesis, chemically mediated, and the landscape connectivity hypothesis. And our team have applied several rules to screen the functional plants. The first one is to use the functional plants attraction or repellence mechanism towards the insects, selecting those plants which can both attract natural enemies and avoid pests. And the second one was to select plants that could provide pollen or nectar to natural enemies, while they have limited benefit to pests. And the third one was to verify the coincidence of the flowering periods with the growth stage of natural enemies so that the phenology could be kept consistent. And the final step was to filter according to the local availability or the cases that had been successfully applied. And in the end, our team has selected 5 out of the 30 functional plants. Then we have conducted further analysis on these 5 selected plants. And the key findings include that there are significant differences in their attraction levels to natural enemies. And the flowering periods of many plants showed a correlation with the activity periods of natural enemies. And in addition, the violet substance of some functional plants exhibited repellent effects on whiteflies. And we also have several application cases in China. And in the following, I will present the cases implemented at Beijing Noya Farm. And this farm is one of companies we will visit tomorrow, and you will know more knowledge about this farm or work from the introduction of Associate Professor Li Shu or Professor Wang Su and others. And the company manages 800 of protected agriculture, and translate to the hectares is about 50 hectares. And within the protected agriculture facilities, there are 43 certified organic products. And we have implied functional plans measurements across various spatial scales, like inside the facilities, between facilities, and within the agriculture park. And between the facilities, we have planted customers as honey plants to attract natural enemies for feeding, which ultimately forms a natural reservoir of the natural enemies. And this approach effectively increased the population of predatory natural enemies, and it has resulted in a 20-fold increase in the predator population. And this measure also leading to 43 reduction in the pest populations. And there was no significant impact on the crop yards. While the use of insecticides crop yards decreased by 34%. And yes, we can see this approach achieves the dual benefits of ecological pest control and stable yards. And we use repellent plants which contain certain volatile chemical molecules to reduce pest spread within the facilities. And by planting means celery and others before the aphids outbreak, and with the timely replenishment, the spread of pests was effectively limited in creating a safer growing environment for the protected vegetables. And we also cultivate mice within the facility, and within the mice act as a banker plant supporting the reproduction of natural enemies. Our research indicates the aphids infection on the maize poses no risk of infection to tomato. And at the same time, the increasing the lady beetles colonization rates by 20%. And this approach can not only delay the pest outbreak by 1 week and also reduce the outbreak peak by 15%. And in a short time, there was an asymmetric interaction between the two aphids species facilitated by lady beetles, and in the long term, the control has been found to be stable. In the whole facilities, we planted roses as the corridor plants to promote the migration of natural enemies and regulate the habitat network. And compared with the control area, the treatment area showed more complex food web structure and higher abundance of natural enemies. And this not only helps natural enemy achieve cross-regional migration, but also effectively regulates the richness and the diversity of regional habitats, and thereby enhancing the overall ecological service function in the farm. And just in a summary, effective functional plants management technology in protected agriculture can improve the monoculture pattern of both within and outside the protected agriculture ecosystem, and they can also break the continuous habitats of pests and thereby optimize the entire ecosystem. Also, it can enhance the sustainable pest control effects of natural enemies and reduce the reliance on From these primary findings, we can observe publication trends over the years in the total publication volume by countries and the collaborative relationship between nations and the authors. And notably, scientists from China and the United States are the primary contributor to publications with close cooperation between the two countries and researchers. And the World of Cloud also reveals the key terms such as nature, plants, and aphids. And prominent authors include Nico, Wang Su, and others. And we also sampled several literatures and extract over 100 items of data, and we have done a primary analysis, and we find that the beneficial effects of functional plants on the predatory or parasitic natural enemies showed significant differences. And our team achievements include the establishing natural enemy production factory, developing habitat regulation technologies for the natural enemy and functional plants, and also pioneering the composition release technology for multiple natural enemies. And these technologies has been implemented across Beijing, Shandong, Hebei, Jiangxi, and other provinces. And this technology also reduce the pesticide usage and achieve economical achievements. The relevant achievements have also gained many awards. And in the future, our teamwork have a series of the work perspective. So I may not discuss this more. And finally, I would like to thank the support of this project, and thanks to our research team and cooperating institutes, as well as the professors, collaborators for their support. Okay, thanks for your attention.
We successfully conclude our seminars. Finally, I think we should give thanks to all the speakers for their insightful presentations. Thank you. Cynthia will give some conference arrangement information. So.
结束了，就我跟他们说所有的安排。Okay, now I think so after 10 minutes we gather at the hall of the restaurant, hotel the hall.